So I'm really pleased to start off today, really pleased to introduce Crystal Chadburn. Crystal is going to officially welcome and open our mini-symposium today. Crystal is a member of our Invasive Species Council BC Board of Directors. She's also a senior invasive plant specialist with the Ministry of Forest Lands, Natural Resource Operations and Rural Development, and co-chair of the Invasive Species Working Group, Interministry Invasive Species Working Group. Thanks very much, Crystal, for joining us today, and I'll just ask you to welcome everyone.
Great. Thank you so much, Jody, and good morning, everyone. On behalf of the Invasive Species Council of BC and its Board of Directors, it's my pleasure to welcome you to today's first virtual Invasive Species Research Mini-Symposium. First, I'd like to start by acknowledging that I'm joining you from the territories of the Coast Salish peoples, and that you are also joining us virtually from traditional territories located across BC and beyond. The Invasive Species Research Conference typically takes place in person, where we're together about every 3 years. Because this year's in-person conference has been postponed due to the pandemic, we decided to offer this virtual half-day, what we're calling mini-symposium, to maintain some momentum and keep us connected. This Invasive Species Research Mini-Symposium is made possible with financial support from the province of British Columbia and the valued input of the Invasive Species Research Advisory Team. So thank you very much to all of you who've been involved. So having planned the research conference almost completely before the global pandemic hit, what turned into a challenge has actually become a bit of an opportunity. That's because by using this online platform and bringing together people virtually, we're actually able to bring together even more people than we would have in person from across the Pacific Northwest and beyond. We have almost 292 people registered for the virtual event, and it looks like we have 168 joined us already, and it's only 9:05, so I expect more people to trickle in throughout the day. So we thank you all for joining us this morning and really hope you find this mini-symposium worthwhile and inspiring. The main goal of our event is to share topical research from within the Pacific Northwest and as well as relevant research from across North America and beyond. Later on, Jody, who started the morning for us, will be reminding us of a document that we have called the Invasive Species Research Roadmap. And she'll also take a few minutes to share a relatively new means to connect and collaborate between practitioners and researchers that's called the Invasive Species Research Hub. So without any further ado, I'll hand it back to Jody to start our formal program, and I hope all of you enjoy the morning. Thanks very much.
Thanks very much, Crystal. I appreciate that. We're so pleased to have so many folks with us today. So before we introduce our first keynote speaker, we wanted to get everybody sort of engaged and started for the day. So we're going to just do a quick poll. And what we're curious about is, what is your relationship to invasive species and their research? So you'll see on the screen now, there's 4 possible answers. We're just asking you to pick the one that's most appropriate for you. So the first, I study or I'm active in invasive species research. Second, I work in invasive species management. 3, I'm interested in invasive species, or 4, oops, what am I doing here? So if you just want to just take a second and just click on the one that's most appropriate for you. I just want to get a sense of, across the participants that have joined us so far, where are sort of what the predominant group is right now in terms of participation. So as Crystal said, one of the main goals of sessions like today is to really provide an opportunity for dialogue between researchers and practitioners across the Pacific Northwest. And so we expect that there's sort of an equal blend of those 2 groups joining us today. So, and I think we're going to have it jump up in the box here too, and we'll see there. Thanks very much, Nick. So what we can see for those folks that have joined us so far today, we've got slightly more individuals that are working in invasive species management, and then sort of an even split between those that are either studying or active in invasive species research or have an interest in that. So, and obviously we respect that some might sort of blend across those categories, but Excellent. Okay, well, thanks very much. So I think it'll be an interesting day today. We're really looking forward to some engaging presentations. One of the things that I wanted to do just quickly before we get into that is also just to get a sense of where everybody's joining us from. So as Crystal said, we're sort of all located across North America, and because we're virtual, we know that we can also include other folks today that might not have been able to travel here. So you'll see on your screen, we're asking that you just open a separate web browser on your phone or your tablet or your computer. You'll see the link at the top, polev.com /ISCBC 198. And if you just click on that link very quickly, you'll see that you can drop in where you're located. So it's a bit of a geography lesson. So the map is North America wide. If you're located beyond North America, you'll see a box in the bottom left-hand corner. You can just click on that. And that'll give us a bit of a representation of where folks are joining us from today. So I can see on my screen that it's starting to be populated. And see where everybody's joining. And we're up to 175 participants that are with us so far today. So this is great. So we'll just give folks a couple minutes to do that. BC is well represented. Many of the states are also represented, which is very exciting. Hello from Mexico, excellent. Perfect, well, this is so interesting to watch. We'll just give it one more minute and see as everybody kind of gets that dropped in, and then we'll see. So again, I mean, one of the great benefits of being able to do this session today virtually is that we can welcome individuals from anywhere to join us. So I just want to welcome everybody, say a personal thank you for taking the time to join us this morning. We're very excited about the speakers that we have for today. Very excited to all leave today having learned something new and hopefully provided an opportunity to connect with some new individuals. So I think that being said, we'll move into our first presentation. So our first session this morning is where invasive species monitoring and genetics meet. We have two keynote speakers that will be with us this morning. So I'm very pleased to introduce our first keynote speaker, Dr. John Darling. John's a senior research biologist with the United States Environmental Protection Agency. His work supports the EPA's program offices by advancing understandings of the risks associated with invasive species in marine and freshwater systems and developing tools aimed at improving the EPA's capacity to respond to those risks. John's also interested in the problem of translating scientific knowledge into appropriate management action. Also, if you're ever interested or in North Carolina and you'd like to do some sea kayaking, John's a certified sea kayak instructor. John's talk this morning is entitled Genetic Methods for Monitoring Aquatic Invasive Species, a Case Study in Rapid Evolution. Thank you so much for joining us this morning, John.
Thanks, Jody. Can you hear me?
Absolutely, yeah, you sound great.
Okay, I'm gonna start sharing. Everyone seeing that?
Yeah, it looks good, John.
Looks great, John.
Okay, great. Yeah, Jody, thanks so much for the introduction, and I really appreciate the invitation to make some comments to start off this meeting. It's too bad we can't be together in person, but I think it's really great that you guys are setting up this virtual thing in the meantime. Yeah, today I want to talk a bit about something that I've been interested in for a while now, and that's genetic methods for monitoring aquatic invasive species. So on my screen, I'm seeing a little window on top of the slides that says the co-host had asked to start your video. Is everyone else seeing that, or?
We'll dismiss that, John, but we're seeing you fine. Thanks for checking.
You're fine? Okay, just wanted to make sure. If any problems come up, just let me know. Okay, in any case, I'm a population geneticist and evolutionary biologist by training. And so I thought that maybe I'd actually start off by using an evolutionary analogy as a theme for this talk. And I want to set up that analogy with a very brief story. and my slides are not advancing. There we go. So this is the cane toad, Bufo marinus, and it's a notorious invasive species. It's been particularly problematic in Australia, where it's become a shining example of how not to do biological control. And there's actually been an incredible amount of research done on cane toads in Australia, a lot of it looking at how invasive populations have evolved rapidly. And it turns out that in relatively few generations, these toads have evolved into invasion machines. So specifically, they've evolved phenotypes that are conducive to rapid dispersal. And as the cane toad population exploded, there were selective pressures at the range margins to develop these physiological and biomechanical traits that allow toads to more effectively colonize unused habitats. And these super toads are better dispersers, and they're more successful at expanding beyond the range limits. But here's where the analogy gets interesting. There are costs associated with this rapid evolution. So it turns out that creating super dispersers results in some unintended consequences. Specifically, these toads are plagued by issues like spinal arthritis and other bone and joint issues. And this poor toad, aside from being dead, had to suffer painful fused spinal vertebrae associated with this hyperinvasive phenotype. So sometimes this is what happens when you evolve rapidly under strong selection pressures. And what do these messed up invasive toads have to do with the story today? Well, that's what the next 25 minutes or so is about, I hope. And I want to start here by talking about the problem of detection. I suspect everyone in the audience has seen some version of this graphic, which shows the invasion curve. And as time progresses on the x-axis, the area occupied by the invader grows dramatically. There are different management actions that are appropriate depending on where you fall on the curve. And what I want to focus your attention on is the little bar at the bottom there, which estimates the return on investment associated with different strategies. And it's pretty universally recognized that prevention is the most cost-effective management strategy. And return on investment falls dramatically in later stages. The problem with the very early stages is that it's really difficult to detect an invader when the population densities are that low. And our ability to do so depends on the quality of detection tools. And so we're always looking for new tools, and there's pretty strong selection pressure on those tools. Obviously, we're looking for tools that are more sensitive, but we're also looking for tools that give us answers more quickly, that are less expensive, and that are more easily deployable in more places. Such tools are a general goal of all biodiversity monitoring, but they're particularly so for invasive species where there's such a premium placed on early detection. But amazingly, a lot of our tools for monitoring biodiversity haven't changed in quite some time. So for example, here's a recent photo of a biodiversity researcher doing fish surveys in a river in Laos. And this is a depiction of what I can only assume is a couple of biodiversity researchers doing basically the same thing in 14th century Iraq. The survey approach takes advantage of technology that is quite literally prehistoric. Of course, there are good reasons that this technology is still in use today. But my point is that by and large, biodiversity surveys still rely on what I'm going to call catch and look technologies, in which we attempt to capture individuals and then we examine them visually to determine their identities. But what if we could do things very differently? What if we could detect species without doing catch and look? How might that change the monitoring world? There are a lot of ways to answer that question, but this graphic shows one of the most ambitious. In a world where we're free from the catch and look paradigm.
Oh, no.
I'm getting, was that a question or just someone unmuted? Okay, sorry. If we free ourselves from this catch and look paradigm, we can imagine a network of devices that assess biodiversity based on analysis of DNA and the ambient environment. Those devices could be spread across the face of the planet, and each of them automatically samples DNA and uses genetic technology to categorize the diversity present and then beams all that information to a cloud-based infrastructure that could analyze the data and infer ecological networks, and even sensitively identify changes in those networks. Those changes could include the arrival of novel diversity that might signal a new invasion, or maybe even shifts in species distributions driven by anthropogenic stressors like climate change. So today's researchers are contemplating a future in which a significant component of diversity monitoring could be freed from the catch and look paradigm, and therefore automated in ways that we couldn't ever before imagine. So if we're on a road towards this new world, where do we currently stand? I want to talk a little about two very different approaches to genetic detection to give you a sense of where these tools are in their development, and to give a couple examples of important challenges that stand between us and adoption and decision-making contexts. And I want to start with environmental DNA, or eDNA, and the targeted detection of non-native species. So the real conceptual advance here can be traced back to 2008 to this paper that described detection of amphibian species by simply looking for DNA in water samples. So this research group didn't try to capture adult specimens. They didn't look for larvae or egg masses. They just grabbed small samples of water and they looked for DNA. And this is now what most people mean when they talk about eDNA. It's DNA collected in an environmental sample without the isolation of the target organism. If we can capture an organism's DNA without capturing the organism itself, and if we can then associate that DNA uniquely with the organism, we have a new paradigm for diversity surveys, something that's very different from catch and look. And the real question for us is whether this approach could actually be used to monitor for aquatic invasive species. And the answer turns out to be yes, although with some caveats. Maybe the most widely discussed early illustration of this was the case of Asian carps in the Great Lakes region. And despite what you might think looking at some of these pictures, these carps are actually very difficult to detect when they're rare. So a group at Notre Dame developed an eDNA method and reported results in this paper in 2011. They were able to detect carp DNA in areas where they had never found fish by traditional means. And most troubling was the fact that DNA was detected in areas much closer to the Great Lakes than anyone thought. And obviously, if there's DNA in the water, there must be fish in the water, right? Well, interestingly, moving beyond the catch and look approach brings along a very obvious criticism. If you're not actually capturing the target organism, how do you know that it's really there? This was definitely an issue in the early days of monitoring for Asian carp. DNA was found in places where people had looked for fish and not found them. And of course, this is to be expected if you have a new tool that's far more sensitive than your old tool. And the speed with which the tool was developed all but guaranteed some unanticipated critical responses. So this is what I'll call the central challenge of eDNA. Is it possible to infer an underlying population distribution from a pattern of DNA detections? And this challenge actually encompasses a number of issues that have been actively researched for the past decade. How long does DNA stick around in the environments? What's the fate and transport of DNA in flowing systems? Can there be alternative sources of DNA besides the presence of live organisms? But the fundamental question is this: can we trust eDNA to tell us where a target organism is? And there's mounting evidence that the answer to this question is yes. So this is a figure published recently by a group of Canadian researchers who have been developing methods for detecting zebra mussels. They tried to detect mussel DNA in water samples pulled from Lake Winnipeg, and they looked at both sites that were known to have a history of invasion, as well as some control sites that were known to be mussel-free. And what they found was that they could consistently detect target DNA at invaded sites, but not at uninvaded sites, except for this site here. That's a float plane dock near the head of the Red River. And it had been noted as a high-risk area because of its location and its usage, but it was considered mussel-free. But after detecting eDNA at this location, subsequent surveys found attached adult mussels at the site. And this is just one of a number of cases, including, by the way, the case of the Asian carp, in which eDNA resulted in early detection and provided valuable information that was unobtainable by other means. And results like these have been demonstrating that eDNA can provide us with important early warnings of the presence of invasive species at population densities so low that they go undetected by conventional means. Other studies have looked at the empirical relationships between patterns of DNA detections and actual site occupancy. These results, looking at 2 amphibian species and comparing eDNA detections to capture rates along river transects, show that whether you're looking at target species density or biomass or proportion of occupied transects, you see a reasonably strong correlation between eDNA and traditional survey results. And a number of research groups are starting to build on studies like these to develop occupancy models based on eDNA detection data. And the approaches have become common enough that software packages are starting to emerge that facilitate the translation of eDNA data into these occupancy models. So the general point I want to make is that a lot of recent research suggests we can address this central challenge of eDNA. We can, in fact, make inferences about where the target species are based on where the DNA is found. And obviously uncertainties remain, and continued research aims to narrow the confidence intervals around these inferences. But it's already clear that eDNA is telling us something, and often something very valuable, about where these species are. But what about this? Can we really deliver on these promises? Most available evidence indicates that eDNA tools, when properly designed and deployed, are indeed more sensitive than traditional tools. But can we be faster and cheaper, and can we deploy more easily? This, I think, represents the next stages in the development of eDNA technology. For instance, there are a number of groups that are working, and in some cases have been working for years, on developing novel ways to sample water for eDNA monitoring. These are all examples that actually exist. Aerial drones for sampling difficult to reach sites, autonomous robotic samplers, in situ robotic sample processors. All of these tools are made possible by release from the catch and look monitoring paradigm. And all of them are moving us towards realizing the ultimate promise of eDNA. Another thing that we have yet to do effectively, I think, is leverage economies of scale. In today's molecular lab, the per sample costs drop precipitously with increasing sample number, assuming you've got the cool robots instead of an undergraduate in a box. Unfortunately, I think we're generally not taking advantage of this fact. Most monitoring programs are processing very few samples relative to the capacity of molecular labs. I think that to truly deliver on the promise of these technologies, we need to find ways of achieving more efficient workflows. In part, this will come naturally as eDNA becomes more popular and there's incentive for laboratories to set themselves up for this. But I think it will also require some real innovation in terms of sample processing. So I said that I'd talk about challenges to adoption of these tools, and I want to turn now to an important one. One of the biggest concerns with end users of eDNA methods is the possibility of false positive detections. But interestingly, it seems that the eDNA community is not entirely clear on how to understand false positives in the context of monitoring. False positives have actually been on people's minds quite a bit recently due to issues associated with coronavirus testing. In fact, there was a recent NPR story in which a researcher in California asked this question: why are false positive tests so common for both zebra mussels and coronavirus? And it's an interesting question, in part, I think, because it's actually misleading. Both the coronavirus tests considered in this piece and the zebra mussel eDNA tests are tests designed to detect nucleic acids. But what happens when there are nucleic acids present, resulting in a legitimate true positive, but there aren't actually live target organisms in the sample? We all know that this is a possibility with eDNA approaches, and yet the default is to refer to such test results as false positives, as is done in this article. And I think that this is actually problematic. Why would you attribute error to a test that is doing exactly what it was designed to do? The error doesn't lie with the test itself, but with our expectations of what the test can clearly tell us. And my concern is that allowing these results to be described as false positives erodes confidence in the method. And I think we need a different way of talking about these situations. There's another reason that there are so many false positives, and that's explained by something called the base rate fallacy. This is a well understood and yet widely overlooked problem in the detection of rare things. This slide shows a classic example using breast cancer screening, but you can see the problem with a straightforward hypothetical example that might be easier. You may think that you have a fantastic method if the false positive rate is only 1 in 1,000 tests. But if you're looking for something that's so rare that you expect to find it in only 1 out of 1,000 samples, then you should expect that half of your positive tests will actually be false ones. Communicating this issue to end users is really important because it emphasizes just how essential it is to have a tolerance for false positive detections when you're looking for something that's rare, as we often are. Even extremely specific tests may deliver high rates of false positives if the base rate is very low. And there's a final issue here related to the high sensitivity of DNA methods relative to their alternatives. Unfortunately, this means that traditional methods may not be a very good way to confirm inference of presence based on a positive eDNA detection. So if I get a positive eDNA hit, and then I go out with a far less sensitive method and get a negative detection, is that a false positive eDNA inference, or is it a false negative inference from the traditional method? And how am I supposed to know the difference? The default, unfortunately, seems to be to trust the traditional tools because we're familiar with them. And I'm not entirely convinced that this is always the best approach. So what does this all mean? It just means that as a community, we're still grappling with how to achieve common understanding of something as fundamentally important as false positive detections. And moving forward, we really need to be clearer in our language and to better educate end users about what they should expect with respect to false positive detections. And these confusions are in part, I think, the result of rapid progress being made in this field, which sometimes seems to outpace our attempts to clearly communicate all of its complexities. I want to turn now to the second major development in genetic monitoring, and that's the emergence of high throughput sequencing. This technology enables generation of massive amounts of DNA sequence data in a short amount of time with little expense. And the key advance in terms of monitoring has been the application of high throughput sequencing to DNA metabarcoding. I believe that all of us are probably familiar by now with the idea of DNA barcodes. They've been around for a while. Put simply, a DNA barcode is a very short sequence of DNA that can be uniquely associated with a particular species. A DNA metabarcode, on the other hand, is a collection of those short sequences that can be uniquely associated with a particular environmental sample. These are generated by extracting DNA from the entire sample, amplifying a barcode locus from all, or at least very many, of the genomes in that sample, and then sequencing all of those barcodes. Those sequences can then be compared again to reference databases to attach taxonomic IDs, giving a full description of the diversity in the sample. And there are two critical characteristics of a barcode. First, a particular sample always yields the same barcode. And second, samples that are different exhibit different barcodes. This is true both of the standard barcoding and metabarcoding. These features are what allow us to use a standard barcode to identify a particular species and to use a metabarcode to identify a particular environmental sample. And recognizing this is important because it enables us to think about using metabarcoding in a different way. We can think of the utility of a metabarcode not so much in terms of whether it's a completely accurate representation of the biodiversity in a sample, but rather whether the imperfect representation it gives us is reproducible and useful. So what do I mean by this? Well, here's an example. These are ordination analyses of metabarcode data from samples collected at several ports around the world. And it's probably not surprising that samples from Chicago look quite different from samples from the Canadian Arctic. And maybe it's not even that surprising that two sets of samples taken from the north and south sides of the island of Singapore look quite different. But we even see distinctions between sets of samples pulled from different locations within the Port of Adelaide in Australia. And what's remarkable and promising is how closely these samples from the same locations resemble each other, suggesting that these metabarcodes are reproducible and that they informatively reflect differences in biodiversity. This approach can be used to recognize changes in community structure, for instance, those associated with invasions. So in these ordinations, metabarcodes distinguish between habitats that either have been invaded by an invasive seaweed, or those are shown in the red circles, or those that remain uninvaded, shown in green. And the distinctions that they see here are apparent for two different metabarcoding loci. And so what's important about these examples is that they illustrate the utility of metabarcodes, even in the absence of any attempt to translate those data into taxonomic information. So there's a first challenge associated with metabarcoding. Can we find standardized methods to generate these metabarcodes that informatively and reproducibly reflect the biodiversity in a sample? A number of recent studies, like those I've been referencing, are demonstrating that we can in fact meet this challenge. But standardization still requires lots of decisions about what types of metabarcodes are most informative and how they might be most reproducibly generated. And of course, we also sometimes want to know about the taxonomy, what species are actually there. So here's an example of results from an analysis of Canadian port samples. The top bar chart shows the distribution of taxa at the phylum level, indicated by different colors, across 4 separate Canadian regions. And the bottom chart shows only those taxa that are known to be non-native to those regions. So taxonomic assignments derived from metabarcodes can be informative regarding the invasion status of these Canadian systems. But assignment to species level can be challenging. So for instance, that study identified 3 sequences present at a site in the Great Lakes that were initially assigned to the European green crab, Carcinus manus, which is a marine species, so that's rather strange. And on more detailed inspection, using this phylogenetic reconstruction approach, they found that there were actually a number of species with sequences identical to C. manus in the reference database, suggesting that the initial assignment was in error. So there's a second challenge of metabarcoding. Can we develop methods that consistently enable confident taxonomic assignments? And the answer to this question is something like maybe or almost. The example I just gave points to challenges associated with the algorithms used for taxonomic assignments. And there are other limitations associated with reference databases and biases associated with different barcode loci, which may be more informative for some groups than for others. So this brings me to a second example of a hurdle to adoption in decision-making contexts. For many years, the research community has suggested that biodiversity studies using metabarcoding have a secondary purpose. They can be used to provide incidental detections of species of concern. These may be invasive species, pathogens, threatened and endangered species, or even species suspected of undergoing climate-induced range shifts. We could use eDNA approaches to target those species, or we could just look for where they show up in these community-scale metabarcoding efforts. But can we trust these detections? Not coincidentally, the answer to this question has a lot to do with false positives. There are plenty of places along the metabarcoding pipeline where a false positive error might be introduced. You might have contamination or exogenous DNA in the fields, contamination in the lab, sequencing error, even erroneous taxonomic assignments. So there are opportunities for species name to pop up in your taxonomy table, even when that species is not actually present. And of course, the community is working to minimize these kinds of errors. But what we might not have recognized, I think, is the risk that this introduces. We live in a world in which end users are hungry for data on the distribution of species, particularly these species of concern. So there's strong selective pressure to develop tools for detecting these species. But at the same time, we live in a world in which the observation of a species in a particular place can sometimes trigger decisions with real implications. So let me give you an example of the problem. Several years ago, some colleagues of mine in New Zealand were doing metabarcoding studies of marine diversity on the North Island. And they generated some species lists using standard quality assurance methods, and were poised to publish when the an advisor working for the Ministry of Primary Industries recognized that one of the species was this parasite of flat oysters. That parasite had recently been discovered on the South Island, resulting in declaration of a controlled area and imposition of limitations on shellfish movements. And the assumption was that it was absent from the North Island, but publication of this list would have changed that. So based on recommendations from the advisor, the researchers went back and conducted some pretty rigorous post hoc QA. And it turns out that the original assignment was actually an error. And it's interesting to consider what might have happened had they published the original list, and if some end user had discovered the species name on it. This kind of risk may be an important unintended consequence of the rapid advances being made in DNA-based biodiversity monitoring. The point here is that monitoring done by metabarcoding is typically done to QA standards that might be acceptable for ecological studies, but are definitely not fit for purpose in the context of biosecurity. So what happens when these data-hungry end users start looking to these lists to check for the presence of species of concern? It's a little tempting to pass the problem off onto the end user with the buyer beware attitude. But as a community, we've long been saying that these incidental detections are a major benefit, and it seems unfair to absolve ourselves of responsibility now. So it appears that we need to separate these two worlds, the one in which biodiversity surveys are done for standard ecological studies, and another in which QA procedures are sufficient to warrant adoption in biosecurity decision-making contexts. Those procedures can be pretty onerous, and it's very unlikely that every metabarcoding study is going to meet them. But those that do can be flagged appropriately so that end users know which studies are fit for purpose in biosecurity contexts, and just as importantly, which ones are not. And in the meantime, it might be wise for practitioners to be cognizant of these issues. So these are just examples of the challenges facing us, and the work needs to be done to meet them. It's pretty obvious that we're some way off from the world depicted here. Note that the vision described in this graphic weds the twin advances of environmental DNA with metabarcoding, sampling divorced from catch and look, coupled with the massive data generation capacity of high throughput sequencing. So quite a bit of effort will be required to achieve this vision of monitoring. But I hope I've convinced you that we're making some progress. And I also hope I've impressed upon you the importance of researchers and end users working together in order to avoid going down unproductive paths. So I want to leave you by returning to our friend the cane toad. It is said that evolution is blind, and strong selective pressures will often result in the proliferation of hitchhiker traits that are less than desirable. Hopefully by now the lesson is clear. Genetic methods for invasive species monitoring have been evolving very rapidly. They've been responding to strong selective pressures for more sensitive and cost-effective surveillance tools. Over the past decade, this may have led to some unintended consequences. But fortunately, moving forward, I want to suggest that we don't have to be blind. We have the opportunity to anticipate some of the challenges and to develop these tools more intentionally. I think that by working collaboratively with communication and foresight, researchers and end users together can create tools that are responsive to management needs and that avoid the undesirable traits that might otherwise have come along for the ride. And with that, I'll finish, and thank you so much for your time and attention, and I'm more than happy to field any questions.
Thank you so much, John. We appreciate your time this morning and really appreciate all the interesting information that you've shared with us. I've seen a number of folks posting questions in the Q&A box, so just a reminder to anybody that had a question come to their mind during the presentation, you can drop it into the Q&A box. You can also vote in the Q&A box. So if somebody's dropped a question that you would love to have answered, just hit the thumbs up button to move it to the top of the queue. So we are going to take about 10 minutes, John. We'll work through the questions that we're able to today. So the first question that we have is, what about terrestrial species? Would eDNA be limited to aquatics, or is it also being applied to terrestrial species?
It's definitely also being applied to terrestrial species. I'll admit that I'm not as familiar with that world. Interestingly, some of the applications that I've seen involve, they still involve water. So for instance, I know that there are groups that have looked at crop pests, and they detect eDNA in runoff from fields to look for the presence of those pests. Other examples, feral swine is one where people are using eDNA in aquatic samples taken from places to look for the presence of feral swine. So in most of the cases I know, the sampling is actually still aquatic, but they're looking for terrestrial species. I suspect there are other examples where people are, testing soils and stuff like that, but I'm just not as familiar with that literature. I think it's easier in water.
Thanks, John. So back to water then. So the next question is, has there been any research done on eDNA detection of invasive aquatic plants?
Yes, definitely. So the degree to which this works has a lot to do with the degree to which your taxon of interest sheds DNA into the water column. And that varies pretty widely depending on the taxonomic group. Some plants are actually quite good at shedding DNA into the water column, and they can be detected pretty easily just by taking water samples and looking for DNA. Certain fish are especially good. Asian carp was a great place to start with eDNA because they're just massive shedders of DNA. Some organisms are more difficult to detect because they just don't shed as much DNA into the environment. But plants are doable, definitely.
Excellent. Thanks, John. The next question is from Chandra. She's asking, metabar coding is often used to assess soil biodiversity. Has anyone attempted to metabarcode to detect invasive plant tissues like pollen, for example, or that you're aware of?
I don't know. People are using metabarcoding for just about everything. So I would not be surprised, but I'm not aware of any specific examples of that.
Great. There's a question about whether this methodology can be used to detect algal blooms.
Yes. In fact, at the EPA, we've done a little bit of work looking at that. We actually looked at some, there was a brief project to look at golden algae in the southeastern United States. So people are using targeted DNA approaches to look for some of these harmful taxa. I'm not sure about cyanobacteria, whether or not that has been applied, but it's certainly a possibility.
Great. For the next question, just asking about sort of the potential for false negatives. And as you referred to, some species that may not shed cells adequately. And so I think the question is, there's still a responsibility to make sure we're sort of adhering to the precautionary principle of, false negative. I'm just trying to find a question in this statement. I think maybe I'll pass. We'll scroll past that one and we'll respond to that one.
So I'm actually glad that was brought up, because a lot of the discussion about error with eDNA focuses on false positives. And I do get the sense that end users are particularly concerned about false positives because of the risks that they bring. But especially in the invasive species world, we know the danger of false negatives. And so I think it's important to balance that discussion of error by considering what it means to have false negative detections. And of course, one of the reasons for going to eDNA is that its increased sensitivity should reduce those false negative detections compared to traditional tools. So thank you to whoever brought that up.
Thanks, Don. We have a question from David wondering, do we know how long zebra mussel DNA can stay in the environment after the mussels have died?
I don't know, and I don't know if it is known. But this is, this is an active area of research, and again, it varies from environment to environment pretty considerably, is what people are finding. And it may even vary from season to season, depending on what's going on in the water column, temperature and pH. So it's a complex issue. I don't know if people have looked specifically at zebra mussels. I'm guessing that probably they have, given the importance of zebra mussels and how much work has gone into detection methods. But I don't know the answer to that question.
Thanks very much, John. I think this is an interesting question that goes further to what you were talking about earlier, is if false positive is somewhat of a misleading term, is there a better label or a different way of describing that?
Well, there was a paper that came out recently talking generally about monitoring, and it made the distinction between observation level false positives and site level false positives. So the observation level would be the DNA test itself. If there is DNA in the sample and you get a positive detection, that's one thing. But if there's no live organism in the actual environments, I think you can think about a false positive at the site level there and still maintain that the test itself was accurate and gave you a true positive. Unfortunately, it's an added complication in terms of communicating, which makes things more difficult. But I think sometimes it's worth it to add that complexity, because I really do, in my experience talking to managers, I see this a lot, where there have been positive eDNA tests and then they can't find the organism. And the default is to refer to that as a false positive eDNA test. And there are a lot of reasons to resist, I think, using that language, some of which I talked about. And my concern is that, and I've seen this happen as well, is that language, there's power to that language, and using that language over and over again really starts to erode the confidence in the method. If you keep talking about these false positives, which really may not be errors from the perspective of the eDNA test, you start to think that the test isn't working, when in fact it may be doing exactly what it was designed to do.
Thanks so much, John. Really appreciate your time this morning, appreciate your presentation, and also your willingness to take questions from the group. So virtual round of applause, and thanks for your time. And thanks to everybody that did raise questions in the Q&A box. For those that we didn't have a chance to address live, we will be taking those questions and working with John to see if he's able to address those offline by email. So stay posted for those. We are going to just take a short 10-minute break now, give everybody a chance to stretch their legs, top up their coffee. I'd ask you to please just keep your Zoom window open. We will be coming back at 10 o'clock, and we will have a timer running on the screen. So please just take 10 minutes and meet us back here at 10 a.m. Thank you. Our beverage of choice this morning. Before we move into our next keynote presentation, we just wanted to take a minute and get to know a little bit more about our participants this morning. So you'll see on your screen a quick poll. For this poll, you can select multiple options, so choose all those that apply. And the question is just, which areas of invasive species work, research, or study are you involved? So there are a number of options: aquatic plants, terrestrial plants, aquatic invertebrates, terrestrial invertebrates, mammals, fish, birds, reptiles and/or amphibians, fungi and/or microorganisms, or all of the above. So we're just curious about kind of where your interests in research and work are aligned right now, just to get a sense of the group that we've got with us. So if you want to just quickly answer that poll, and then we'll pop up the results on the screen in just a second. We can get a sense of what everybody's involved in. think I personally find easier when you can choose multiple options. You don't have to choose just one. Perfect. And I see some folks are answering in the chat box. That works as well. If you want to be involved in the poll results, just click it right on the poll screen itself. Thanks very much, Nick. So you'll see the results have come up across the group. So very heavy towards involvement with terrestrial plants and aquatic plants, sort of an equal mix around aquatic and terrestrial invertebrates, and also around reptiles, amphibians, and fish. So it looks like we've got some folks that are working across all ranges of these species groups. So thanks very much for participating. Hopefully today gives you an opportunity to sort of connect with other individuals that are also working in similar areas. So thanks so much for participating in the poll. Our next keynote speaker is Dr. John Gaskin. John is a supervisory research biologist with the United States Department of Agriculture's Research Service in Montana. For the last 18 years, the focus of John's research has been on population genetics and molecular systematics of invasive plants. Invasive taxa of interest include salt cedar, rush skeleton weed, hawkweeds, pepperweeds, Russian olive, and teasel. John is American, but secretly wishes he was Canadian. John's talk is entitled Using Genetics to Inform Invasive Weed Management. Thanks so much for joining us this morning, John.
Hi. Hey, thanks, Jody. Can you guys hear me okay?
Yeah, your sound is great.
All right. I just wanted to, first of all, thank you very much for letting me come and talk here. I'm going to share my screen here. Tell me when you can see that okay.
Yeah, it looks great. We can see it now.
Great. Well, I'm happy to be here because I love talking about weeds. I love thinking about genetics of weeds, what DNA can tell us about weeds. And so that's kind of the title of my talk right there, Using Genetics to Inform Invasive Weed Management. You know, and I'm going to talk about a few species. Let me see if I can get everything advancing okay. Because, you know, there are things that DNA can help us answer that you can't always know about a plant just by looking at it. You know, we often need to know what species is this weed. You got to know what it is if you're going to control it. How does it reproduce? The plants vary a lot in the ways they reproduce, and if you want to target making it not reproduce as much, you have to understand that. That helps us design some control options. Are there different genotypes or lineages within a species? You know, and as we know, there's variation in species, and they might vary in the ways that they resist or tolerate our control. And how are these genotypes or maybe some lineages distributed? Is the invasion in the eastern US the same as maybe in the western US? And is that the same as what's going on way up north in Canada? And where do these invasions come from? I'm not a biocontrol person, but I work with some, and I play one on TV, as they say, and they often need to find some host-specific enemies, some things that have co-evolved over a long term with the species to be an effective biocontrol agent. So we have to go find where they were co-evolving so we can find those insects or biocontrol agents. And then it comes down to, okay, you want to learn something more about a plant. What DNA method or marker are you going to utilize? And John very nicely talked about eDNA. It sort of fits in there. That's an answering a presence or absence question. Sometimes we want to know relationships, and sometimes we want to know more about the species and what's going on within it and how it functions, basically. So I'm just showing here that we can either look at slowly evolving DNA if we want to compare things across kingdoms. It would have to be something that hasn't evolved too quick that we've sort of lost the message. In the middle, you can see that maybe we want to look at some moderately fast evolving DNA if we want to tell the difference between species. This is where barcoding would fall in about in that range. But often when I'm interested in an invasion or someone is, we need to look at some really quickly evolving DNA so that we can tell what are the differences between individuals in the group. So we might look at microsatellites, which are little repeats of DNA that mutate really quickly. I mean, over a few generations, often there'll be a bad replication of the DNA, and that'll leave a little mutation for us. Or something a little more high throughput like RAD-seq, where we're taking, you know, thousands of samples, putting them on a plate, we're tagging them so we know what they are, and we're getting a ton of information out of that, and that's very efficient. And or something a little more simpler, a little more old school like AFLP. I'll have some examples where we're amplifying the DNA that we've got from it, we're chopping it up, and we're seeing how big the pieces are. And the pattern of how big the pieces are can tell us about those plants right there and how they're related. So the first one I want to talk about is flowering rush. And I apologize that Canada is excluded from my map as usual. Sorry. But flowering rush, I think most of you know about it because it's in BC. And it's spreading throughout Western North America quite a bit right now. It's a big problem in Montana for us right now. And it has effects on the habitat, native fish habitat especially. And we're thinking that there are, you know, some differences in the flowering rush that's running around here in North America, and maybe some differences in how you might be able to control it. So the DNA analysis questions that basically have come up from people is things like, what ploidy is this invasion? There are diploids of this plant, and there are triploids. So some have 2 sets of chromosomes like we do, some have 3 sets of the chromosomes, 3 copies. So are they a little different? And what genotypes might exist in North America, and where are they distributed? And where did they come from? I mean, flowering rush is from Europe and Asia. And you can see a list of countries there. And we're not even sure if we know exactly where else it might be from or where it might have been moved to. And it's sort of a pretty ornamental plant that people like to put in places. So we already know some stuff. This is the good thing. We know a lot about a lot of these plants, but we don't know everything we need to know. There were studies that focused on Eastern North America where sort of awareness of that invasion came on quicker, or the invasion was going on more rapidly over in the East. We know that there might be changes from being diploid to being triploid that are rather recent and reoccurring in Europe. So these triploids might keep evolving out of diploid populations. And we know that most natives are triploid over in Eurasia, and most invasives over here in North America are diploid, or at least that's what Kleiber and Eckert showed us back in 2005, which is really cool. And one thing we know that both diploid and triploid North American populations are usually clonal, meaning their DNA of their children is the same as the mother, even though the diploid seed's viable. So this thing's making seed, but it's not any different from what its parents were. It's kind of weird. So a little back into Europe, you can see a map here on the top left of Europe, and you can see that most everything is triangles, which would be some triploids. And here they were counting chromosomes basically, where they were looking at stomates and making a count. And down in North America from this earlier study, they were focusing, of course, over here in the Great Lakes and on the St. Lawrence, and almost everything there would be the round circles would be diploids. They did a little surveying out here. They looked at a plant in Montana. They didn't even put you guys on the map, but I don't blame them. They're worried about the East Coast. That's the Eastern US. That's what they were looking at. So that tells us a little something about what diploid and triploid genotypes are running around. But maybe within either the category of diploid or triploid, we can get a little more information. So we're gonna go a little more fine scale, and we're gonna look at some AFLPs, these amplified fragment length polymorphisms, and try and answer these questions. So, you know, people kindly sent us samples from Pacific Northwest and for Europe, where we're looking for matches for biocontrol, and from Midwestern and Eastern USA, so we can sort of compare our invasion to theirs. And a few kind of weird things showed up. This is a scatter plot of the genotypes, and if they're more similar, the dots are closer, and if they're more genetically distant, the dots are further away between any pair of dots. And the clear circles are native samples, and the orange circles are North American samples. And as you can see, there are a lot of clear samples. There are a lot of genotypes running around, and it says their country next to them and how many plants were sampled for that population. And then we see that we have some orange dots. Here's British Columbia, and it's similar to plants that were from Slovakia. Here's some Illinois plants, and they're similar to some things from Hungary and something found in a European nursery, and so on. We have some invasives from the East and Midwest down here. But what's really wacky is this is Alberta, British Columbia, Washington, Idaho, Montana, Minnesota, Wisconsin, Michigan, 159, a ton of plants, all have this genotype out here that was not similar to anything we found in the native range. That's frustrating. We obviously don't know the origins of this most invasive, or at least most populous genotype that we have here in North America. So given that, how are these distributed? Well, that popular one is showing up as these blue teardrops here. And you can see around in your area in BC, there was this genotype 1. This is the most populous one. And up at Bucci Lake, there's a genotype 2. But you can see that we have sort of a population structure going on here. Out in the east, we mostly have this genotype 4 that's green. But then we see a lot of the genotype blue as we go further west out there. But certainly they're out in the east also. So a little bit of distribution of different genotypes in different areas. So a little side note, sometimes we get things and we genotype them, and they come out as some weird genotype we've never seen before in AFLPs. And we go, oh, well, do we have another Butomus genotype? Or maybe not. So a nice sort of check that we do is then we go back and we'll sequence the DNA on those in a marker that we can use to compare across species instead of comparing within a species. And it turns out that, yeah, some of these genotypes that we would look at, when you sequence them, most of them come out as matching Butomus exactly for the DNA marker, a chloroplast marker we're using. But some came out to be actually something else, Sparganium, which I'm assuming, yes, looks quite a lot like our flowering rush if it's not flowering and it's just a green little stick showing up, a leaf showing up out of the water. So a good thing to check, and a DNA tool that can help us verify things there. So what about if they're diploid or triploid? How do we look into that? Well, we usually do things like looking at flow cytometry, where we can shove a bunch of cells through a little thing and it looks at them and it tells how much nuclear content's in them, which is kind of fun and kind of easy. You need fresh tissue. Or sometimes you can also look at the morphology of the stomate and see that the triploid ones are a little different shape. But a little side note on that, so basically we could see a diploid at a certain length here on the x-axis, and a triploid we would expect to be 1.5 times further down on the axis, and that works pretty well. And sometimes we get ones that, it's a crummy little peak and we don't know what it is. Maybe it was stringy tissue or the sample was rotting. But basically, if we're having problems, we can even fall back to the old school way, which you may have done in school, where you're squashing things and putting them under a microscope and counting chromosomes, which people who are really quite good at that were able to do for us. And we can go back and verify whether we're looking at something that has 26 chromosomes, which would be a diploid, or 39, which would be a triploid. So that's pretty useful. So what do we have in terms of ploidy? Well, these blue common genotype 1 in the West seem to be all triploid, but we did find a diploid of it, which is a little weird, and I'm intrigued by that. But out at Bucci Lake, we found something that's a triploid, but a different genotype. But basically, we're looking at mostly triploids in the West, and then these purple diploids in the East. So who cares about ploidy? Do you, if you're sitting there and you want to control it, if you want to manage it? Well, different ploidies act differently sometimes in plants. The diploids are better. They can make seeds well. Triploids often have some chromosomal arrangement. The chromosomes can't get together, and mitosis, meiosis, all those things, and they don't make good seeds. Just as some hybrids don't make good seeds in this world. But that's saying if you have diploids, assuming they can make They can make good seeds and triploids can't. They can only reproduce maybe by root fragments or something else coming off of it. Are they differently invasive? I don't know. Haven't done that test. And does it matter for controlling them? Whether we're using a chemical control, a manual control, probably not. You can rip one out just as easily as the other. Biological control, I don't know. I don't know the answer to that. But what we do know so far, Europe has a ton of genotypes. North America has a few, and that's pretty typical when you get an invasion. There's just a little bit of the genotypes come over. And what's weird is not all the European populations are clonal, and there's a lot of different genotypes if I looked in a lake. But here in North America, it's usually one genotype in a lake, and it doesn't vary. That's weird. I don't know why. I'd like to know why, even though these things might be putting out seed and outcrossing. We have diploid and triploid genotype 1. That scares me a little bit. Think about that. Does it matter? I'm not sure. And we haven't found a close match for a very common genotype 1. That bothers me because they're actively working on biocontrol of this thing, and they need to find something that will attack it. And we don't know where it came from. CABI and Army Corps of Engineers will or do have all the genotypes, so they can do testing of potential agents. And they can do other tests. So this is kind of useful to give them all the genotype diversity that is in the North America. It's not that much, it's just a few genotypes. So one thing that's a little wacky that we want to know about genotypes, here's an example, is if we're looking at biocontrol with a foreign disease, which CABI in the UK was looking at and still is looking at. Will the disease, if we bring it over, attack all the Butomus? You would hope so, because there's a risk to biocontrol, and you want to make sure that it's effective. But the funny thing is, the rust, which was a smut, this picture on the right, does not attack genotype 1, which we supplied them with. We said, here it is, grow it in the UK, try your disease on it. Didn't like it at all. It would attack the other ones. So that would be kind of a waste for us to bring this disease over and try and develop as a biocontrol if that's our most common genotype running around in the Pacific Northwest. So still looking for a match of where it came from. Maybe then we could find a disease or an agent that'll attack it. Another agent they're looking at is a little weevil that likes to swim underwater and go attack these things. And I don't think it cares what genotype it is. Another interesting thing is, well, what about disease? Sometimes we rely on, diseases that are just hanging out in North America that might attack something like flowering rush. And in this case, it was a little interesting. There's this black bar showing triploids. The x-axis is your latitude that it came from. Your y-axis is proportion of disease incidence, how much it gets hit by diseases. And these were grown in a common garden. So we collected genotypes from everywhere. They took them to a common garden, they grew them, and they saw, well, how many things are attacking it? Is it relatively resistant or not? And it turns out that in general, the diploids, this gray bar, had more incidence of disease. So that's the East Coast plants. And our plants, the triploid, had less incidence of disease. Okay, that tells us that's not so great for us in general. But also it mattered by latitude, what latitude these plants came from. They were grown in a common garden, like I said, but they came from different latitudes. And the further north they were from for the triploid, the less they got attacked down in a common garden. And the further north for the diploids, the more they got attacked. So you can see there's a lot of variation in the way these things might get managed. And it's nice to know what they are and how things are attacking them. A few caveats on this. I mean, there's a lot of questions. I mean, just because it gets hit by a disease doesn't even mean it might damage it anyway or slow it down. But it's interesting nonetheless. So a little side note, I always like to point out that plant invasions are rarely homogeneous entities. There could be a lot of different phenotypes. You've seen a lot of morphological variation. Plants are great at that. And you've seen a lot of, there might be a lot of genotypes in an invasion. This is rush skeleton weed from an earlier study I did. And you can tell by the colors that there are different genotypes in different places. And, these evolutionary processes that we have out there, selection, drift, what reproductive mode it's using, founding events when it was brought over and moved, these shape this structure and the diversity of an invasion. And knowing that can help inform us about and help guide weed management. Turns out there are certain genotypes of this rush skeleton weed that do get attacked by a mite or do get attacked by a fungus, and the others don't. And you'd want to know that, what you've got, before you start deploying your biocontrol agents. Let me shift to another one. This is Verbascum thapsus, common mullein. Once again, I apologize, but I'm pretty sure it's up in Canada too. I've seen it up there. And this thing's invasive in obviously all across the US here and up in Canada. And I worked with some fine people down at Colorado State University and out in France on this project. But this goes in the sort of category of how do weeds reproduce? I'm intrigued by this. They can outcross? Yeah. Make seed. They can self make seed. They can be apomictic, like rush skeleton weed, where they don't even need pollen to make a seed. Virgin Mary kind of thing going on there. Are they just clonal? Like a lot of aquatic plants are very clonal. And I've shown you some examples like in Butomus, there's a lot of clonal reproduction going on there. And this affects their population structure eventually as they're invading. And it might affect our weed control method. So, interesting thing about this Verbascum, it's really showy. Everyone can see that, knows it's around because they can see this tall yellow spike that's as tall as we are. And thing is that Donnelly showed that it's fully self-compatible. So it can self and make seed. It doesn't need another plant around. There wasn't any inbreeding depression. It was okay with selfing, but it has a mixed mating system with this reproductive strategy that depends on pollinator availability. So the tall plants were seen by the pollinators and they did a lot of outcrossing. The shorter plants aren't found as quickly by the pollinators. And after a day, the flower kind of gives up and says, well, no one pollinated me. I'm just going to self. Okay. And I'll make some seeds anyway, and it'll be fine. So that's kind of cool. But why would you be so showy? Why would you put so much energy into this tall spike of big old flowers if you don't even need or use sex to make seeds? A question I think about. So we looked at a bunch of plants, and we, in the USA and Eurasia for this species, I can see 400 something in the US and about 500 in Eurasia. And I was very interested, well, how many genotypes do we see? And it turns out 32 in the US, that's not very many, and a lot in Eurasia. So you can look at sort of a genotype over number of samples ratio here, and you can see that in the USA, wow, way lower. We don't have very many genotypes running around. And in the native range, yeah, tons of genotypes running around. So a lot more diversity in the native range. And that's kind of expected when you look at things like founder effects. But actually, this ratio means that there's not a lot of outcrossing going on in North America. So that is going to leave a pattern that we can see. And here is a map. And the colors are different genotypes, red being genotype 1, green being genotype 2. And each of these circles is a population of about 10 plants or more. And we color-coded them for what genotype's in them. And some interesting things you find out when you're looking at this fine scale is, first of all, half of these invasive populations were monotypic. They only had one genotype in them. This is a showy outcrossing thing that bees and bumblebees and other things love to move the pollen around. So what the heck's happening there? Why are these things monotypic? And we only found 32 genotypes, like I said, in North America. Same genotype, this red one, can be found across the range of the invasion. That's really weird. From up here in Washington, way down into Arizona, over to New Mexico, this is the same exact genotype as far as we can tell. And we have pretty good resolution with AFLPs. Why aren't there more genotypes? What is it? Is it all founding effect that just a few genotypes were brought over to North America? Well, that might have something to do with it. Or are they just selfing all the time? It's very possible that that's really driving this. There's a lot of selfing going on. They are not getting the pollination they need to outcross. So that gives implications for biocontrol a little bit, but not as much as saying well, where did these guys come from? So using these same AFLP or DNA markers, we found the highest genetic similarity between the USA invasion and plants that were from Belgium, Germany, and France. So this is nice. And some of these were like 99% matches. The genotype's the same over there in Europe. But what it tells us is there are a lot of places, including all over here, over to near, is that Azerbaijan? Maybe so. Or Spain, where our genotypes did not come from. There's a lot of range where they didn't come from. But what this information is very nice. We can say, you're interested in controlling genotype 1? Well, if there's some host specificity at a fine scale, like some insect might like some genotypes and not others. Doesn't always happen, but it does happen. And you want a genotype 1 to control it, well, they came from this population. Exactly, we could tell you latitude and longitude, go over there and look around and you may find insects, and some of those might be host specific to that. Some might just be general insects that eat anything. Some might be insects you could find throughout the range. But this really helps us narrow down where a thing came from. And this saves a lot of time for the people looking into biocontrol. So, like I said, what can we do with this? There's also an unsupported suggestion. This reminds me that common mullein has some herbicide resistance or tolerance. They looked at this a while ago. So if you're spraying this thing to control it, okay, we've seen herbicide tolerance. We see this popping up in agricultural weeds all the time, the evolution of things that are Roundup resistant and things like that. But it could happen in more wild situations too, where you're spraying and controlling. So with this, we have identified, and we could supply pretty much all or the very common genotypes to someone who is doing herbicide resistance research. We have been able to identify specific origins for searching for these co-evolved biocontrol agents in the native range. That's pretty helpful. Let me go to number 3. And my number 3 topic is, what thistle is this? You know, I'm a botanist. I'm not great at identifying plants, but you know, I often run into things where I go, what is this? Or it doesn't quite key out the way I think it should when I'm looking at my key and looking at the plant and going, I don't know what that is. It isn't what the key says it should be. So a good group of people, mostly Oregon Department of Ag people, worked with me on this project. And what they found was they were looking around and they were looking at thistles, and they honestly thought, this kind of looks like something, but we're not sure. Let me go back though. I'm gonna back up for a little refresher course for Carduus in North America. They're all non-native. They're all from Europe and Asia. Most of them are state noxious in some state or province. You have Carduus nutans for you people who work on plants. I apologize to people who don't work on plants, but you have to watch this anyway. Carduus nutans has this big head, you know, and it nods there. Easy to tell. Some other ones, Acanthoides and Crispus, they have sort of round involucres or heads right here. These are sort of round. And then you get to ones like Tenuiflorus and Pycnocephalus, which have these sort of cylindrical heads. They're not as round. And that's how you can tell them apart. And you can sort of see their distribution. I think you've got some or most of them in BC. These are USDA maps, and they're not entirely accurate, but they're pretty good. But they were walking around in Hell's Canyon in Oregon, Idaho, that border right there up under Washington, and they were noticing, we have something that we're not sure what it is, and it's growing in these remote, hard to get to areas. And sort of a little map here of where they found this weird looking plant along the Snake River here in the US. And they thought, okay, well, we know it's not these big-headed or round-headed things. It looks like something that is got a cylindrical head here or something. Oh, hey, it looks a lot like Carduus pycnocephalus. That's Italian thistle. He said, looks a lot like that. Well, wait a minute. But the heads are on these longer peduncles usually. It's more loose. The heads are sort of loose instead of compacted, as you see here, into groups of 2, 3, 4, 5. So they said, okay, what is it? It's probably a Carduus. Well, when you can't tell from morphology, and they looked at all the keys in Europe, and you can't really tell very well from the keys either, but they said, okay, we can't tell, so let's look at some DNA. So here we're just doing some typical DNA sequencing of a nuclear region. It's pretty easy, and the fact is, you know, there's DNA from a lot of different plant species in like GenBank, where you can go match it on your computer and go, oh, okay, that's what it's most similar to. So what they looked at when they sent all the samples to us, we sequenced all the ones that are weird morphologies, and they were all identical in this sort of tree here, this phylogeny or dendrogram that we have here. They were all identical to each other, mostly, that's good. But the one they thought it was closest to, this Carduus pycnocephalus, this Italian thistle, is up here in the tree. And it's, okay, and here are a bunch of US samples, and we even got some samples from its native range in Italy, steal some samples off of GenBank. They're up here. The other Carduus are sort of here in the middle. And then we get some Carduus from Europe and Asia, another one from Europe and Asia, from Turkey, and then, but this thing's here. It's not closely related to the ones up here. There's like, 4 or 5% DNA difference, which is a big difference in DNA for this marker. So what the heck is it? Okay, it's not Carduus pycnocephalus. It's not Italian thistle, but I'll tell you, that is a garbage can species. They throw a lot of different morphologies in Europe and Asia into that thing, and they say, yeah, we think those are all the same species. But everyone's had they've been suspecting for a long time that they were wrong. It was kind of anything's in there, maybe things that aren't so closely related. So is the thing they found a hybrid of something? Well, no. I mean, we looked at nuclear DNA, and that is DNA that's mother and father combined, and it's overlapping. So we could see, well, okay, does it have a lot of shared markers between two species we already know about? No, it didn't have that kind of overlap of DNA at any of the critical loci. So it's not that. Is it an Eastern European Italian thistle? Well, Italy's west. This is Eastern Europe. Well, maybe it's close. They even found some samples that I found on GenBank, Carduus pycnocephalus, that's Italian thistle, but it's the subspecies from Turkey. So not very Italian there, but some more Eastern things. Okay, yeah, that's what it had identical DNA to. So, but I've already said that's a really bad name. There's a better name, and if you're into botanical rules and laws, the actual name that it should be is Carduus cinereus, and that is Turkish thistle. So they've got to go revise the genus because everything's messed up. So that's the problem for us botanists. We've got to revise that genus to reflect its evolutionary relationships instead of just its morphologies. But we're calling it Turkish thistle. Is it invasive? I don't know. We're sort of at that new stage of finding something and not knowing a lot about it. But what we did do is, okay, we took a modified key out of the Flora North America, and one of the co-authors did the Flora North America, so yeah, he knows what he's doing there. And we can key this out, and we can say, okay, if you get down to the point where the heads are sort of cylindrical, these are usually pedunculate, meaning a little more loosely clustered, and that's how you can tell it apart, Turkish from Italian thistle. But where is it? Like I said, is it invasive? Is this a problem? We're at that, John showed us the chart of, okay, if we can control something in the beginning of when we're just finding it before it gets widespread, our cost-benefit ratio is going to be way better. But is it a problem? I don't know. So here, the green is where Italian thistle's found down in the US. I'm thinking it's also up there, but here's Turkish thistle they found just here. So not very widespread at this point. Hard to get to in Hell's Canyon, but not very widespread. Is Turkish thistle in BC? I don't know. This is a really good opportunity for me, though, to ask you all, maybe to keep your eyes open for it. If you see it, I mean, you have various ones. I'd say it's not likely that it's up there right now, but you know what? No, it might have been in Idaho and in Oregon for decades, and people were just assuming it was Italian thistle and not differentiating it from Turkish thistle. But now we know what to look for. So keep an eye out for this morphology, if you will, because DNA is one thing, but we can't do eDNA on this sucker. It's not in the water. So we're going to rely on people looking and doing our sampling that way, sadly. So Italian thistle, like I said, the heads are in some clusters that are tight. This isn't the peduncle for an individual head, but here there are short ones there that are all compressed, and Turkish thistle over here. And look at the peduncle length when the head's mature. It's going to sort of be stretching out as the head gets more mature. When it's very young, they still might be tightly clustered. And here's some exciting pictures of how it can bloom at a very young age and a very short morphology, or it can be pretty tall up there where you more expect it to be a few feet tall, a meter tall or more. So that's all I had to say, but I did want to say that, every time I give a talk, someone comes up and asks me a really good question that leads to a research question usually. You guys who are listening, especially the managers in the field, you're the ones who see weird things going on or have questions about things and how to control them. And that's what I really enjoy there. So with that, I will stop here, and it looks like we probably have some time for some questions.
Thanks so much, John. I love your passion and enthusiasm for the topic. It's great to see, and I think we've all learned a lot during your presentation, and also have some new things to watch out for, which is great. So we're happy to help with that. We will take probably about 5 minutes for questions. So if you haven't already dropped a question in the Q&A box, feel free to jump in there and drop your question. If you like a question that's there, go ahead and like it, and it'll up in the queue. So we'll just start from the top. The first question, John, is does flowering rush have mycorrhiza associations that you're aware of?
That's a great question. I have no idea. And I've been to a few flowering rush meetings, and I haven't heard it discussed. Yeah, that can be pretty interesting. You would think sometimes these non-native things don't have great mycorrhizal associations unless they brought them with them, because the mycorrhizae sometimes are picky, some are pretty picky, some are a little more general, but no, I don't know. That's a good question. I'm going to look into that one too.
Great. The second question, and we know you don't know the answer to this, but is there any speculation around where genotype 1 of flowering rush came from? Could it be newly evolved in North America?
That's a good question too. So genotype 1 of flowering rush, the thing we have mostly in the Pacific Northwest, is really genetically distinct. I would think if it evolved from one of the other genotypes we had here, I don't think we've had enough time here. I mean, we're talking that would take a really long time to get that many mutations if we look at its mutational rate in general. So I'm thinking, and other people are thinking, of course, we just haven't gone to the right places. So they have been looking for samples out of China, Japan, sort of southeast Asia, as far south as that thing goes. We've just had, it's been a little troublesome getting the samples from out of there. And now we're trying to get herbarium samples that are like at the Kew Herbarium in the UK. They have samples from over in Asia, and we're going to take them and get the DNA out of them, which is a little easier than traveling, especially right now. We're not going over to Asia right now. I was supposed to be in Canada now for something else for collecting, and Canada did not want me. I don't blame them.
Well, we'll look forward to welcoming you at a different date in the future. Another question from April is, have you looked at the cultural significance of mullein as part of the spread? It's considered a plant of cultural significance to some Métis communities in Canada. Just wondering if that's something that's been addressed or looked at in your research.
Was that flowering rush?
No, mullein.
Oh, for mullein as a plant of cultural significance. That's pretty interesting. I did not know that. That's really interesting to learn, because it came over a long time ago. This is one of those that came over hundreds of years ago from Europe and Asia. It was used as a pesticide, something to kill fish, I've heard, which makes me not want to eat it. But repeat the question. Have I looked, do I think there's any cultural significance tied to its spread? Is that where we're going?
Yeah, so the question, sorry, we've moved it off the list now, but I think the question was just, have you looked at the cultural significance of the species? And maybe, John, that's a better one to think a little bit on and address by email after.
Yeah, because my answer is no, but I would love to learn more about the cultural significance, because yeah, often if we learn how people were using this or thinking about this plant, we can learn a little bit more about how it spread, and then we can do a little better modeling and predicting of where else it might go.
Excellent. We've got time for one more question, so let's take this last one from Chandra. She's asking, how do we determine which evolutionary processes are mechanistically involved in different stages of plant invasion, and why do we need to understand those processes for management?
Oh, Chandra, I know you. That's a good question. I want to see that question there.
If you prefer to see it so you could look at it, John, you can just click on the Q&A box at the bottom of your screen. Yeah, it's in the open questions. You'll be able to.
It's in the opens, okay.
Yeah, or I think Nick may have just moved it to.
Got moved.
Yeah.
Is it in the answered box? But
yes, it's at the bottom of the answered box.
Well, I want to address that later, or maybe talk with Chandra, or I'll reply in an email about that one, because we're often really I'm not even looking at like functional genomics of what these genes mean and what it might mean in their period of evolution of their invasion. But it's a really good question to look at it. That's just a whole nother direction that is super important.
Excellent. Thanks so much, John. Hugely appreciate your time and your energy this morning, and your willingness to answer questions from the group as well. And so we'll welcome you back for the panel a little bit later on this morning. So thanks very much for your presentation.
Thanks.
And thanks to everybody in the group for the excellent questions too. It's great to see so many coming from different folks in the group. So, so for our next discussion item, in addition to this event and our traditional research conference, we wanted to just take a couple of minutes and talk a little bit about a few other initiatives that our Provincial Invasive Species Council of BC has been involved in. And so we'll just, I think, John, will just ask you to stop sharing your screen, and then we should be good to move forward. Perfect. So the first, so the initiatives I want to talk about are really talking about facilitating communication and collaboration between researchers and practitioners across the Pacific Northwest. This is something that our council has been talking about, and partners across the Pacific Northwest have been talking about for a number of years. And how can we use those conversations to help direct the future of invasive species management and prevention in BC and also beyond. The first initiative that I want to talk a little bit about is the Invasive Species Research Hub for the Pacific Northwest, which was developed by the Invasive Species Council for BC with the financial support of the province of BC, and also with a large network of research advisors that supported on the project. So a huge thanks to all those who have been involved. For those of you that aren't familiar, the hub was launched last year in 2019, and the real purpose behind the hub is to facilitate connections between researchers and practitioners across the Pacific Northwest. So you'll see you have an option of choosing that. Nick's doing a sort of a live demo for us as we go through this. So one of the key features of the hub is a searchable database. Membership at the hub is free. Approved members can search the database and choose to post their own profiles in the hub. We'll go through a bit of a live demo. So Nick has searched for knotweed. So you'll see an example of searching using the word knotweed. 2 profile summaries came up. By clicking on the profile, the full profile displays. And so this example, you can see Dr. Nicholas Wong, my colleague, is displaying the search results. So it includes research papers, areas of interest, and more. And it also allows members to connect and discuss research-related topics of their choice using an online communication platform called Slack. So if members are active on Slack, their member Slack handle is included in their profile. And so here's just a quick look at Slack, just so you can see some of the communication capabilities that you have if you choose to engage in the Research Hub. So discussion threads are listed in the left-hand panel. Members can decide which discussions they want to follow and participate in, as well as create their own discussion threads if there's something they're interested in talking about that hasn't been raised yet. So I really just want to encourage individuals to join the Research Hub if they're interested. It's an excellent way to connect and stay engaged with current research, and also raise new research topics and ideas if there's something that you'd like to bring to the research community. So it's a really interesting way to have a sort of a two-way dialogue. We will include the link to access the Research Hub in the chat box. So you'll see that Ali's popped that in there now. And certainly would invite you to explore that and look at it a little bit more detail for yourself and sign up if you're interested. As I said, membership is free. So it's certainly something you can try and see what your interest is. The second resource that I wanted to talk a little bit about is the research roadmap. So, which is the collective vision of the requirements and priorities for research on invasive species management and prevention in BC. So the first invasive species research roadmap was developed in 2009 before we hosted our first ever research forum in 2010. We also hosted a second session in 2017 and did a facilitated dialogue at that session to update the roadmap and to take new input on priorities. So really, we wanted to just let everybody know that there is an updated roadmap. We're just finalizing it with our advisors group, and then we'll have it available and out for individuals to provide input and comment on. So I just wanted to give you a quick highlight of the key themes and indicators that are included. The first is management tools. And so you can see this on your screen, new management methods for priority invasive species to be developed and widely accessible. The second theme is best management practices, and the indicator of success would be best management practices for priority species and industries are available for all regions in BC. And again, this is a BC specific document. The third theme is ecological restoration, with an indicator of success being an increased number of restoration projects across the province and sharing of strategies and successes in terms of what's worked well with restoration work. The fourth theme is pathways of invasion and vectors of spread, which is a common discussion when we're talking about invasive species. The indicator of success would be identifying and reducing new pathways of invasion, and also identifying strategies for each of those pathways to prevent further invasive species spread. The fifth key theme is biology and ecology of invasive species. And the indicator there is just an improved ability to predict and manage invasive species introduction and spread. And the sixth, and certainly not last, theme is the social and economic impact of invasive species. Certainly, we also are looking at the environmental impact, but this theme is speaking more to the social and economic impact, which isn't always as well understood. And the indicator of success there is just a greater variety of cost-benefit analysis of those social, cultural, and economic impacts of invasive species in BC, accessible to those of us that are working so that we can better communicate the need for invasive species management. So I just wanted to share that information. As I said, the updated research roadmap will be provided and posted to the Invasive Species Council BC website in the next month or so. We can certainly notify folks when that's live and available. And certainly look to your input on that and hope that you'll find that an interesting and useful resource in your work. Moving on, that takes us to our second break of the morning. I just want to again so much thank John and John for your presentations this morning. Very engaging, very technical topics that you delivered in such an easy and comfortable way. So I really appreciate you taking the time to do that. We are going to take a 10-minute break again, so we'll ask everybody to come back at 11 a.m. Again, please just keep your Zoom active and grab a stretch break and update your, top up your coffee if you need to, and we'll be back at 11 a.m. Okay, thanks very much. Northwest perspective. So we have a series of 3 speakers joining us who are actively doing research in the Pacific Northwest for a series of 3 shorter presentations. We will have time a few minutes after each presentation to answer questions with each speaker. So certainly do drop any questions you have within the Q&A box as we've been doing. And then following our 3 speakers, we'll ask all of our speakers to come back for a plenary discussion at the end of our, to wrap up our session today. So I'd like to introduce our first speaker of this session, Dr. Shawna Lee Chai. Shauna Lee is a senior research scientist with Innotech Alberta. She graduated with a PhD in plant sciences from the University of Cambridge, where she was a Bill and Melinda Gates Scholar. She's conducted significant research on invasive species issues over the past 15 years. First in Jamaica, where she researched the invasive white-tailed deer and implemented invasive plant control programs. And now in Alberta, where she conducts invasive species risk assessments and needs assessments for the Alberta government. In her spare time, Shauna loves cooking and playing board games. Welcome, Shauna.
Thank you so much, Jody. Let's share my screen. Okay, so good afternoon from Alberta. I know Alberta isn't exactly part of the Pacific Northwest, but we're neighbors. And it's a good reminder that for invasive species control, it's very much a neighborhood issue, and it requires a couple countries or states and provinces coming together around the issue to really solve it. So on my first slide here, I have a picture of Kelowna and in British Columbia. And when I think of Kelowna, I think of the mild, hospitable climate. And it occurred to me that several places in the Pacific Northwest will become even milder and more hospitable under climate change. And so that means mildness and habitats that will be created for even more pests and other types of invasive species. So I thought it would be useful to touch on some of the management considerations around invasive species for the Pacific Northwest. And I think other speakers in this session will go into further details about particular issues. So I'll just start by giving you a broad overview. So when I think about climate change, I remember that climate is one of the major influencing factors for pest and disease and other invasive species distribution. So when I think of the climate changing, I think of the pest species complement and the invasive species complement also changing. So that's, I think stochasticity is the name of the game when we're thinking about managing invasive species under climate change. We have to really expect the unexpected, and we have to be prepared to respond to the unexpected. I think tools such as risk assessments will help us to do this efficiently, sort of bringing to the fore all of the literature that has been built up around a particular species, getting to know what it looks like, and sort of readying yourself for it to come in and for you to deal with it. Triage will also be important. We're going to have to decide whether we're going to manage a species actively, if we're just going to let it be and sort of tolerate it, or if we're going to accept it and make use of some of the values it brings. From climate change, we can very much expect new climate matches between the Pacific Northwest and certain other areas of the globe. The picture of the map I have here shows Alberta and the various climate matches between Alberta and the rest of the globe. And you'll see that between the two time periods, even a place as harsh as Alberta that goes to -40 degrees and lower in the winter, it has increased climate match between the historical climate data and the projected climate change time period. So Alberta will match up with a greater number of countries, and so it can tolerate species from a greater number of countries. Using expanded spatial scales and spatial lenses will be important, as well as expanded temporal lenses. In the Pacific Northwest, we can no longer just focus on our province or our state, and we can't really focus on 2020. We very much have to broaden that out. Look at what's coming up from the southwestern states. Look at what's going to be on the horizon in 10 years' time, 5 years' time. So we can have enough time to bring everyone up to speed and ready ourselves in various ways. When I think about climate change, I also think about timing mismatches for control. I think we've developed our control methods, be it spraying or mechanical means. We've developed them over the past couple decades and really come to rely heavily on them. And climate change is going to turn some of that on its head a little bit. Things might need to be sprayed 2 weeks earlier, 2 weeks later, and we have to be open to that. And then the other issue that I'm sort of interested in, and I wanted to bring it to your attention, is this issue of sleeper weeds. So we've used agronomic species pretty liberally to provide ground cover and in reclamation. And by and large, many of them have stayed to where they've been planted, but in an altered climate, I think some of them are going to start encroaching on other areas. So I think an essential strategy in managing invasive species under climate change is drawing close to your neighbors and networks, which is very much what we're doing here today in BC and with the Pacific Northwest. Open those lines of communication. Once they're open, build those relationships, establish a cadence. I like how this seminar is held once every 3 years. Gather intelligence from federal bodies, the CFIA, from provinces, from neighboring states, from places like China. And don't be afraid to use experts from outside of your province or your state, because that's where many of these species are originating. And it's in all likelihood experts that reside where those species are from are more knowledgeable about best practices and how to deal with particular things and how they look. I wanted to mention something on strategic planning. Sure, you're going to hold true to your anchor functions, your mandate of whatever organization you represent. But I would encourage you to apply that climate change lens to everything that you do. So from your broad strategies to your individual tactics and activities, really hold them up to the climate change lens and ask yourself, what extra added thing can I do to make sure that this is robust in an era of climate change? And of course, when you plan strategically, you will have a good handle on what your invasive species threats are, what regulatory tools are at your disposal. And I would also really encourage networking and engagement of whatever clients you serve, your staff members, and your influential stakeholders to make sure that everyone is on the same page, your staff is familiar with the direction you're going in, and your influential stakeholders, very often your funders, know the importance of what you're doing. I view the Pacific Northwest as a region that is very much on the front lines of lots of things. And depending on how you perform or not, you certainly protect the interior of Canadian provinces and US states. So it's a major transshipment port you have there in the Pacific Northwest. You're the link to the whole other side of the world. Many areas have a mild, inviting climate. And we know that climate change will result in this poleward expansion of species. So things will be moving up, so be mindful of that. And when folks think of climate change, I think they think of this gradual sort of welcoming warming, but really it's the extreme weather events that occur that really can shake things up. So think of fires and floods. Those knock back native communities or agricultural communities, and they provide the opportunity or the niche space for new species, novel species to come in, settle, and establish themselves. And really, the stakes are high for the Pacific Northwest. I give an example here of the agri-food sector in British Columbia. This is an industry that has thousands of small companies, small to medium-sized companies. The sales and the exports are in the billions of dollars. the industry provides jobs for thousands of workers. And when you really look at it, one pest can wipe out an entire trade link and wipe out and disrupt so much of our economy and the health of our society. And for my last slide, I will leave you with this one. 2020 is the year of COVID, but it's also the year of plant health. The FAO has designated 2020 the International Year of Plant Health. And I very much like this because it signals to me that the world is standing together around the issue of plant health. We're not alone. The UN year is around raising awareness of how protecting plant health can really impact some big issues in our society. Things like ending hunger, reducing poverty, protecting the environment while still having economic development. And it highlights some really interesting stats, like 40% of food crops globally are lost to pests annually. And you and I are in a position to do something about that. It also brings to bear the fact that protecting plant health is so much cheaper than dealing with a plant health emergency. And so it provides that business case for our work. And it talks about the threats to plant health, including trade and travel, and of course, climate change, which is the subject of our session today. So I think especially in a year where COVID is top of mind and protecting domestic food supply chains is also top of mind. And so it's wonderful that this is the year of plant health so that issues like plant health aren't lost in our emergency issue that we're facing today. So I'll end with that and I'll take any questions you might have.
Thank you so much, Shauna Lee. We very much appreciate your time this morning. I will invite participants to raise any questions in the Q&A box, and we'll just raise them as we go. If somebody's dropped a question that you like, feel free to give it a thumbs up to move it to the top of the list. So Shauna, our first question is, what are your top research priorities related to invasive species and climate change in the Pacific Northwest?
Yeah, I did a study a couple of years back. It was for Alberta, and it was looking at some of the new invasive plant species that were really high priority, high risk, and the likelihood of them establishing in Alberta. And so I think with David Clements out of Trinity Western University, we're very much interested in replicating that study for British Columbia. And so that's my biggest sort of research thing that I'm looking forward to at the moment.
Excellent. Thank you, Shauna Lee. And I think that wraps up the time we have for questions. So I just, again, a virtual thank you for your presentation this morning. And we'll invite you back to join us for the panel after our next two speakers.
Thank you.
Thank you very much. Next, I'd like to introduce Dr. Angela Strecker. Angela is a director of the Institute for Watershed Studies and associate professor in the Department of Environmental Sciences at Western Washington University. Prior to that, Angela was faculty in the Department of Environmental Science and Management at Portland State University and the director of the Center for Lakes and Reservoirs. She's also an associate editor of the Journal of Aquatic Sciences, and in her spare time enjoys open water swimming, canoeing, and kayaking. So welcome, Angela. Thanks for joining us today.
Thanks for the invitation. Can you hear me okay?
Yes, your sound is great.
Great. Okay. Thanks so much for the kind invitation to be here, and I'm really excited to talk about some aspects of climate change and how it affects aquatic invasive species. So the invasion pathway is a really commonly used framework to think about how invasions are going to, all the different steps in the invasion that affect invasions essentially. And climate change is going to have impacts at all along this process. And so I'm going to highlight a couple of examples that have kind of direct application for freshwater environments. And so first, if we think about pathways, we know that the shipping lanes in the Arctic are going to become much more open for ship traffic. And so this diagram is showing all the different routes that are going to become available by the year 2050. This is greatly going to change in the introduction of species from distant places in many places along these routes and at the terminal ends. When we think about the next set of filters, so environmental constraints, obviously climate is going to have big impacts there. So a study that was done in Ontario looked at a number of fish species and how their distribution had changed over 3 decades. And they found that almost all non-native species had shifted northward in just a pretty short period of time. This is a picture of bluegill that had moved almost a degree of latitude northward and had increased its occupancy by about 50%. So clear that a number of species are going to be able to spread to new habitats. When we think about emergent consequences, so the kind of the next bottom phase here and how that impacts with management, I want to illustrate an example. I actually helped collect some of the data from this, and this was a study in the John Day River in Oregon. And this is looking at the spread of smallmouth bass in black. So this is the river. And so upriver is up here and downriver is down here. And the white circles are juvenile Chinook salmon. And what we see is that smallmouth bass are pushing upwards into this habitat. And then we kind of see that there's this zone of overlap where they're both present. But temperature really highly regulates this zone of overlap. And we know that if we're thinking about, for salmon, riparian restoration to manage water temperatures, that we really need to be thinking about where Chinook salmon are going to be in the future, but also where smallmouth bass are going to be in the future because of this, this kind of intentional non-overlap. And so all those kind of factors are going to come together. And so we need to have a better understanding of how these different factors work together. And to do this, we first need to know what we know and what we don't know. And unfortunately, there's a lot that we don't know. So this is a really recent study that did a literature review over 400 studies. And of that, they only found 3 studies that had observed impacts of climate on aquatic invasive species in the Pacific Northwest. And all of those were on fish species. So this tells us that there's a lot that we don't know yet about where the species might be and what their impacts might be in our region. So it's predicted that there are going to be fairly serious consequences for native species. And this is a nice kind of illustration that climate change is going to have both direct effects on species of conservation concern by altering the conditions in water, as well as the availability of water. But it's also going to have indirect effects that are mediated by invasive species. And these can be things like changes in predation and competition, increased disease transmission, as well as changes in habitat that are enhanced by invasive species. So one of the ways, and this is pretty common in the invasion kind of pathway literature, is to think about filters that control establishment of invasive species. And we can use these abiotic filters to try to predict which species, may be able to establish in new places in the future. And so here's an example with some kind of common invasive species and some of the filters that are that control their distribution and establishment. And that we know that climate change is going to alter some of these filters. So for example, warmer temperatures, less winter hypoxia, and so on. And so this is a useful first start in terms of understanding which species are going to go through the filter. So these are the species in the bottom that make it through all the filters. But what we don't understand is how, for instance, some of these variables are going to interact together. And so potentially, if there are interactions that are unpredictable, we might get more species passing through these filters, or conversely, fewer species may pass through these filters because of these interactions. The other concern is that it fails to incorporate biotic variables, and so that we know organisms interact with each other. And so this is kind of a gap again in our knowledge and this framework of trying to predict which species are going to move with climate. This is another study from Ontario that looked at the importance of both abiotic and biotic factors in influencing establishment of fish at the northern range limits. And they looked at several species, but I'm just showing the panel over here for largemouth bass. And essentially, we see that there are a number of abiotic factors that are important, having largely to do with temperature, but that there are also some biotic factors, such as the presence of other centrarchid species, the presence of predators, as well as the diversity of their prey that also influenced their establishment. So clearly, both of these factors need to be considered. So I want to switch gears quickly and conclude by talking about some of the common approaches that are used, and I'll illustrate some of my own work in these areas. And so we can kind of divide these up into empirical studies. So for example, experiments that attempt to manipulate temperature and see how different species respond. Surveys, so that example from Ontario with surveying the same lakes over periods of time to see how species have spread. Models, looking at, for example, process models that look at mechanisms, or species distribution models. And so I want to take a quick pause and talk about, it's not necessarily a dichotomy that should we, are one of these better than the other. It's all of the above, I think, are very important for us to understand the effects of climate on invasive species. So the first study I want to highlight was work that was funded by Bonneville Power Administration, and what we were trying to understand was whether zebra mussel veligers were controlled by a combination of temperature and oxygen. And so this is this idea that variables interact together to influence invasive species. And so we surveyed zebra mussels in a reservoir in California. And in the winter, of course, we found none, which is not surprising. As the year progressed and the summer temperatures got warmer, we saw that there's most of these different life stages of veligers, of zebra mussels, are in the upper mixed layer, as indicated by this dash bar. Although we do find some layers beneath this, but we find nothing below the anoxic depth, which wasn't surprising. And then further into the summer, we see that kind of same pattern where the anoxia and mixed layers kind of at the bottom. But there also seems to be an upper limit, and that's probably controlled by temperature. And so studies like that just kind of demonstrate, what is the biology of different species. Another approach is to use a process model, and this is an example of work that was also funded by Bonneville Power, and it was to try to understand the factors, if we can model the factors that influence mussels. So thinking about the inputs, things like oxygen, organic matter and algae, and the outputs, and how temperature is going to affect all of these processes. And then we can scale that model, for example, up to a reservoir or a series of reservoirs, and think about how attached mussels reproduce to produce juvenile stages that are in the water column and could be transported. And so models like this are really important to try to understand the fundamentals of species biology in specific systems. Next, species distribution models. And so what these models do are they make a model of what the contemporary distribution would be if we had perfect knowledge of a species based on environmental conditions, and then uses climate models to predict where species would be in the future, so simulating future range shifts. And this is work that I worked on in the lower Colorado River Basin down here. And this is a model for desert sucker, which shows the darker colors show the places where it is most likely to occur. And what we found with this, with the future range shifts, we found that overall non-native species increase their range sizes, what we would call climate change winners, whereas natives decrease their range sizes, what we would call the climate change losers. But importantly, most of these types of models, which are also sometimes called environmental niche models, fail to account for how species interact together, including the study that I took part in. And so this is kind of a failing of these types of models that really needs to be improved in the future. And the other concern is that there are just very few of these types of studies in aquatic systems. And so with this review, we found that there are hundreds of publications that use these types of models, but for the most part, they're in terrestrial systems and they're on plants or invertebrates. And so there's really a big knowledge gap there in terms of how species may shift in the future. So I'll conclude by just saying that, again, highlighting the knowledge gaps that there are in terms of thinking about how AIS will respond to climate change. And overall, I think that my concern is that we just need a more mechanistic framework that thinks about how climate change will have diverse effects on AIS. And lastly, we need to provide managers and stakeholders with really robust predictions about the spread and effects of invasive species in order to give the best information. So with that, I think I probably talked for long enough, and I'm happy to answer any questions.
Thanks so much, Angela. You were right on time, so not to worry. Thanks very much for your presentation and for sharing your wisdom with us. I just want to remind folks that if you do have a question for Angela, you can drop it in the Q&A box. I don't see any in there yet, so maybe what I'll do, Angela, is just ask you the same question that we just raised is, from your perspective, or maybe in your research, what are your top research priorities related to invasive species and climate change?
Well, I think, yeah, I touched on some of these. I think we just don't necessarily even know the most fundamental things about species biology. We don't know, what are their upper thermal limits, nor do we know also what their lower thermal limits are. So I did some work on the aquarium trade, and what we found is that we have very little information about a lot of the species that are in the trade, and therefore we don't even know if they're going to be able to establish in places that are mild, like the Pacific Northwest. And so I think we need more basic biology. And again, we need to understand how these factors are going to interact together. So for example, changes in temperature and flow regime, how are those going to interact to affect species?
Excellent. And we do have a question that's been raised by the group is, do you have any suggestions on how to fill these knowledge gaps or how to address these knowledge gaps?
I'm a researcher at heart, and I think we just need more investment in basic science. So I think there's always a push to want to do more and more novel research, but in reality, we just don't even have some of the basic building blocks of knowledge that we need right now to make robust predictions and to do the models. And we also need to know more about the distributions of species, where things are, so that we can build better and better models. But again, not a lot of funding is available for just basic monitoring work. And so, those are the types of studies that we need to really build and make better predictions.
Excellent. Thanks, Angela. We have one last question from the group is, what about plasticity, where species evolution changes the limits of where they can invade?
So important. What a great question. Yeah, we know that for some species, they have huge amount of plasticity, and they're just going to really be able to track conditions really well. But I would say that we know this maybe for some species that are well studied, but I think there's a lot of organisms that we don't know how plastic they are. We don't know how well they're going to be able to track. And so I think that's a really big knowledge gap. So more of the types of studies where we test the limits of their ability to respond.
Excellent. And we did just have a question from the group is, could you just maybe define the term plasticity for those that might not be familiar with what we're talking about?
Sure. Yeah. So that's essentially the ability of an organism to respond to different conditions. And so, if the temperature changes, can it survive in that different temperature? It's a little bit complicated. Yeah. I'll leave it at that.
Excellent. Thanks so much, Angela, for your time this morning and sharing so much great information. And also thanks for taking the questions from our group. We'll ask you to come back for the panel after our next speaker.
Of course.
Perfect. I'd like to introduce Dr. David Ensing. Dave is a plant ecologist with Agriculture and Agri-Food Canada, who specializes in understanding ecological and evolutionary mechanisms underlying invasive species. He's bringing his expertise, or has brought his expertise to studying invasive and native plant populations across southern BC. He's joining us today from Banff, Alberta, but is soon moving to Summerland, BC. When he's not working, Dave spends his time hiking and camping in the backcountry. Welcome, Dave.
Thanks very much. Just to get the screen share going here for a second. Good, so hopefully everyone can see that and hear me all right.
Yeah, very good.
Excellent. So thanks everybody for the invitation, for attending my presentation today. I'm going to be talking a little bit about drilling into one of the ideas here about problems controlling invasive species as going forward under climate change. And unfortunately, it ties back reasonably well into some of the talks we've been hearing about today. So the first thing I'm going to say is that I've created a little box up here in the top right where you can put my face if you want me to be involved in your presentation today. Hopefully that should mean that you don't overlap too much with any of the stuff I'm showing you today. The second thing I want to acknowledge is that the work I'm presenting to you today and talking to you a little bit about is some ideas that I've developed kind of through my career, but more recently with Tyler Nelson and Dr. Chandra Moffat. We were all together working at the Summerland Research and Development Center with a focus on invasive species in general, but talking from my perspective on invasive weeds. And finally, to acknowledge the extensive support of the BC Ministry of Forest Lands, Natural Resource Operations, and Rural Development, which I'll probably call FLNRO if I ever have to refer to it again today. So I thought I'd kick things off today with this photo of us working in the field, as it allows me to not only reminisce about times when we were actually allowed to do field work, but also to set you in the world that kind of our developing research program finds itself these days, and that is rangeland weed biocontrol. But since COVID has restricted our field work this summer, I thought I'd talk to you a bit about the ideas we've been developing in lieu of spending time out in the field. That links well with my background as a plant evolutionary ecologist, interested in species distributions and the factors that limit their further spread. One of the most obvious things an ecologist like myself can tell you, of course, is that species geographic distributions occur along three sort of main axes of environmental variation: latitude, longitude, and elevation, and that can be seen in this map of mean annual temperature of North America. As any incipient species spreads outwards, or as a novel introduction spreads outwards, it will eventually reach some sort of range limit, and these range limits are often defined by climate, particularly towards northern latitudes or higher elevations. Tight relations between climate and species distributions mean that we can use environmental variables to predict the spatial extent of invasive species, as we have done using ecological niche models or species distribution models, as Angela was referring to them earlier, as we've done with invasive hawkweeds across the Pacific Northwest. Built into any species invasion, though, like this hawkweed spreading across the Pacific Northwest, are species range expansions. And when a species expands its distribution across latitude or elevation, individuals and populations encounter changing climatic variables, and in particular, changing season lengths. Faced with this challenge, species must respond through one or both of two kind of primary mechanisms: plasticity, as Angela was just talking about, or the flexibility, I guess, of a single genotype to express different phenotypes when it encounters different environments, or the evolutionary adjustment of population trait means. Indeed, the investigation of plastic and genetic control of traits in response to spatially varying climate as invasives colonize new territory can yield insights into how they might be predicted to respond under temporal variation in climate that might happen under climate change. For instance, and in what is becoming something of a classic system, purple loosestrife has rapidly colonized a steep latitudinal gradient in season length across eastern North America that reaches an abrupt limit in northern Ontario, somewhere around Timmins. But the question can be asked then, what limits this range expansion at that point? So first thing to show is some evidence about that local adaptation. So if you look at the top panels on the figures I'm showing you here, and again, this is for Lithium salicaria or purple loosestrife, we can see that in each common garden, whether you're in the north, mid, or southern latitudes, of the local genotypes indicated by the color of the points matching the color of the background is the most fit. That is, so southern populations produce the most offspring in the southern location, and similarly, northern populations or northern genotypes produce the most offspring in the north. Such reciprocal transplants are required for understanding adaptation to variable environment, and as you can see again, that these local genotypes exhibit the highest fitness. The other thing I want to note here is, though, is an overall decline in fitness as we approach that upper or northern elevation or northern latitude in the limit. And critically, this pattern is a result of declining season length towards that upper limit that imposes strong selection on the phenology, that is the timing of important life history events like reproduction, in this case of plants, flowering time. We might think of emergence time for insects or things like that. In order to reproduce quick enough in the short growing season available at the northern populations, individuals must reproduce early, which also means they do so at a smaller size, and as this decline would indicate, therefore produce somewhat fewer offspring. And the opposite pattern is true of the southern portion of the species distribution. So purple loosestrife has tight relations between locally adapted genotypes and the climate environment in which they exist. This has happened relatively rapidly since introduction to North America. This is encouraging for species threatened by climate change, but maybe less so for species interactions. And again, these are themes that have been coming through today's talks. And this is especially true in the rate of, as the rate of anthropogenic change might be faster than individual species can respond adaptively to the changing environments. So as I said, some species are expected to respond to climate change differently. And crucially, responses to climate change may vary among interacting species, which can lead to mismatches among those species with serious consequences for each. So I'm going to go through a couple of examples here. So on the x-axis here, we just have the date of something occurring. And on the y-axis, we have the proportional occurrence of putatively interacting species. A migratory species like the plover shown here will probably be more sensitive to mismatches, and mismatches more likely to occur if the resource it interacts with, in this case, this fly species over here, has a narrow bell-shaped distribution. A narrow temporal distribution increases the probability that any interaction will be completely lost in a given year. For instance, if you were a late arriving plover, you might miss out on this food resource altogether. Such patterns are widely recognized in the case of migratory birds and their food resources, whether these are plant or animal-based, and have been well documented in migratory birds that breed in the Arctic and at high elevation. They're also common between plants and their pollinators. In this case, as climate change has advanced snowmelt, as we're going towards the left on this x-axis down here, there's an increasing mismatch between peak flowering time, or flowering onset in the case here, shown by the green line, and the emergence of those bumblebee pollinators that are associated with this species. So for example, if the emergence time of the flies over on the left here, or the flowering time of the Corydalis plant species over here on the right are short and narrow, they will not be a good resource for birds arriving too late or bees emerging in the spring too late for those flowers. Correspondingly, especially in the case of the Corydalis here, it's not going to receive the pollinator service in early snowmelt years. So these examples are of tightly linked phenologies across trophic levels, and we're wondering about how this might play out in a similarly tight relations in biocontrol. Suppose we have an increasing climate change on the x-axis, which you could also consider time, and a species phenology on the y-axis here. For instance, as climate becomes warmer, we might expect an insect control agent to adjust its life history. Perhaps it emerges from winter dormancy sooner as snowpack is reduced and soil warms earlier in the year. If the target, in this case an invasive plant, does not respond as rapidly or maybe to the changing climate, we might expect that an increasing mismatch between their timing results in a decline in control of these species. And in an extreme case, this could lead to a rebounding of a previously controlled invasive by a specific control agent. So given that there's been some murmuring of population rebounds in some important biocontrol systems, we are interested in to what extent this phenomenon has been investigated in the biocontrol context. So we conducted a literature review using search terms like phenology, asynchrony, biocontrol, biological control, and all their sorts of interactions, and conducted forward and backward searches of the citing literature we found. We looked at how these varied through time, and we found that, not surprisingly, we could see a peak in literature being reported through the 80s and into the current time. Now, this is probably true of pretty much any topic. If you have garbage cans, it would probably look something similar. We're interested in patterns more specifically of different systems. And so we're interested in biocontrol of both insect and plant targets separately. So breaking those down, this is the pattern we see. Nearly half the studies conducted are general review topics or ideas papers. 36% of studies, and this is much more consistent through time, were focused on insect targets. And this is often talked about in terms of asynchrony between the predator or a parasitoid of a target insect. Plant targets, what I'm calling here, which we might consider weeds, they are scattered somewhat haphazardly earlier on and have picked up some interest lately. But crucially, none of these plant studies, and I haven't gone through all the insect ones to state this, but none of these have been documented or indeed been interested in a climate change induced mismatch. So these are the kinds of ideas that we're interested in studying in Summerland when we're looking at two well-established biocontrol systems, that is spotted knapweed on the top and St. John's wort on the bottom, and how their population dynamics have shifted through time under their biocontrol programs, and in particular, if climate change might alter these systems and result in potential population rebalance. So again, I just want to highlight that this, of course, is not done on my own. I want to thank Chandra and Tyler for their input on all this work, and of course, the support of some really wonderful co-op students throughout the past year that I've spent at Agriculture and Agri-Food Canada. And again, just to reiterate my thanks to FLNRO, particularly Val Miller and Susan Turner, as well as collaborators at Agriculture and Agri-Food Canada in Lethbridge. And thank you for your time.
Thanks so much, Dave, for your presentation. Very much appreciated. Just a reminder to folks to drop any questions that you might have in the Q&A box. We do have one there, but it's meant for the panel, so we'll stick to the next one. So the question then, Dave, is what are your thoughts with regards to the strength of St. John's wort populations in Southeast BC?
Yes, this is a new system that we're kind of getting into. We spent most of the past year working on spotted knapweed. There is definitely some indication that St. John's wort has been rebounding. One of the key things that we want to get into is whether or not that's actually true. So these are really old systems. People that are around today, it's like more than 50 years of biocontrol in some of these cases. People around today might not have known how bad the invasions were in the past. And so the question is whether or not, this sort of cyclical nature of biocontrol, where we have essentially predator-prey interactions going on, where you expect some flux in the abundance of the target from year to year. If we're just seeing lately, we're in some of these higher fluxes of the target, and then it'll eventually drop back down again, or if we're actually seeing what is actually a rebound to levels not seen in quite some time. If it's the latter, then we're going to get into the mechanisms and potentially, I know there's some other people working on some ideas about that, and what I propose today is maybe something that could be looked at also.
Excellent. Thanks very much, Dave. I'm not seeing other questions in the chat box. Oh, wait a sec, I spoke too soon. So the last question then, we'll just take this last one, is how long do the seeds of those two species remain viable in the soil? If that's a question you know.
Great question. I don't know. It's something we're really interested in, with particular note, spotted knapweed. So it's, we're talking about ploidy levels before, which is like the number of copies of DNA in any individual. The spotted knapweed in North America are tetraploid, pretty well all of them it seems. And the individual plants themselves live much longer than the diploid diffuse knapweed or the diploid spotted in the native range. And so they produce a lot of seeds. We're really interested in that seed production. They only reproduce that way. And so we're really interested in getting after the seed bank a little more carefully. So there's been some good studies in Montana showing that a seed bank threshold level that might be indicative of successful control. And so we're interested to see if we're just waiting still for spotted knapweed to actually crash out. And so that will depend on how long those seeds are viable in the soil. And so that's a really long way of saying, no, I actually have no idea how long they last.
Excellent. Thanks so much, Dave. Really appreciate your presentation and your time this morning. I'm going to ask you to stay on camera, and I'm going to invite our other presenters from today to join us for plenary. So John Darling, John Gaskin, Shauna Leachai, and Angela Strecker, if you'd like to rejoin us. Just a reminder for folks, if you'd like to see videos of all of the speakers right now, you might want to choose gallery view at the top of your screen, just so you can see everybody. And also a reminder that if you haven't already, You can certainly drop questions meant for the entire panel into the question and answer box at this time. So we have a few questions that we're going to start with, and then we'll start working through questions from the floor as well. So welcome back to our speakers. Thanks so much again for rejoining us. The first question I wanted to put to the group, and I know, Shauna Lee, you've answered this already, but I did just want to put to the group a bit of a question around what your top research priorities are related to invasive species and climate change in the Pacific Northwest. And so I was hoping I could go to each of you for maybe your top 1 or 2 priorities on that topic. And I think I'll just start top of my screen, Dave, if you're willing to start.
Sure, yeah. If everyone's not tired of hearing my voice yet. So I think there's a couple of things going on here. My talk highlighted one of them right there, which is the potential rebounds of previously controlled species as climate change shifts both their distributions into areas where maybe there will be a lag between when the agents can. So like, let's say spotted knapweed expands northward in response to climate. How long will it take the agents to follow and exert control on those novel populations? And then looking into mechanisms for why we might see a mismatch or something like that in a given location, not looking at the spread. And I think the other thing I was thinking of is, especially from a Canadian perspective, there's a lot of invasive species that are problematic outside of our region that we can expect to be coming soon. And so I think touching on what Shauna Lee was suggesting, interactions with our collaborators, especially to the south, and it's really great that we have so many people from the States here today, to look at what problems are they recognizing and how might future climates in our areas match up to what they're experiencing, and we can learn from what they've seen so far.
Excellent. Thanks so much, Dave. I have to be formal now because we have 2 Johns in our panel. So Dr. Gaskin, your thoughts?
You know what really sort of interests me and part of our group down here at ARS in Sydney is when it comes to climate change, we're really interested in how that's going to affect biocontrol, because, you know, a climate change is going to affect an annual plant, its distribution somewhat. You know, perennial plants maybe a little slower, but the insects, the agents that we want to use to control those are much more sensitive to this interaction with the climate, I would think. In like an example is with salt cedar down in Wyoming, we have Diarabda that we use for a biocontrol, and it'll have multiple generations, you know, 1, 2, or maybe 3 even in a season. And in the Diarabda, if they can get that extra season to come out and defoliate the plants, it can really hammer. It can really hammer things and do a better job of controlling salt cedar. But they take this risk if it's a warmer, if we're getting more degree days for it to develop. They take this risk of, okay, I'm going to try that 3rd generation. I'm not going to diapause down in the soil. I'm going to go for it, and I'm going to come out of the soil and make another generation. But then you get a hard freeze because it's pretty risky to go late into the season, and it'll kill off a large portion of the agent population, and they won't be able to get going next year very well. So we really want to know a lot more about doing better biocontrol, and like Shauna Lee said, use that lens of climate change as we're looking at things. Like in leafy spurge, we also want to be looking at, we don't know enough about the agents. We don't know enough about what they can tolerate, and we don't know enough on how an increase in temperature or other factors might really give them an advantage, or, you know, make it so that their predators are having a big advantage. So we got all these trophic levels going on, and it's pretty complicated. And I agree with Shauna Lee. We need more people just studying and understanding the basics of how all these organisms exist and evolve.
Excellent. Thanks very much, John. Angela, I'll go to you.
Yeah, that's a great question. And I think that the interaction of the environmental variables, I think that's a really big unknown, is that we know temperature is going to change, but it's not the only variable that's going to change. And that has different impacts depending on what your organismal needs are. And so understanding how those variables are going to interact together, where there's going to be a synergistic, so greater than expected impact or less than expected. I think we just lack a fundamental understanding of that for a lot of species.
Thanks very much, Angela. Dr. Darling.
Yeah, so one of the things that I work on at the EPA is ballast water related risk assessment. And so Angela earlier mentioned the opening of these Arctic passages to vessel traffic. Things like that are going to be a big deal in the future, I think, and trying to forecast how that's going to impact the arrival of new biota to different regions in the Arctic and also outside the Arctic that are receiving shipping through those passages, I think is going to be interesting. And it's not just the opening of those passages that's making changes either. It's things like changing energy markets associated with climate change that can really influence where things are coming from and where they're going to. And there's some interesting research being done looking at these economies and how they might influence pathways of invasive species in the future.
Excellent. Thanks very much, John. Shaunalee, did you have any further thoughts you wanted to offer on that question?
Yeah, I would just add to what John said about ensuring we have our control means prepped and ready to go. So biocontrol, pesticides, manual control, because this is something that could really affect core issues like availability of food. And there are just so many issues around what makes our civilization successful that could be turned on its head from the issue of pests and climate change.
Absolutely. Thanks very much for your thoughts on that question. We do have a question that's been raised by Audrey from the group. So she's asking, is there a way that iNaturalist or a similar citizen science platform could be used to help support gaps in understanding the interconnectivity between invasive species and native species? And that's a question to the panel. So I'll maybe just put that out there if there's somebody that would like to address that question. If you'd like to read the question as we go through it, you can see it in the top of the Q&A box in the open questions. We have a volunteer.
I'll jump in for a sec. I've used an app for sort of community science oriented things when I do my PhD work. And so I think something like iNaturalist could work. I'm not actually that familiar with iNaturalist itself, but I was looking for phenological measurements of a plant I was studying, and people were able to report to me whether or not the ones they encountered were flowering or not flowering and that kind of thing. And I found it quite useful. It was hard to get a lot of uptake. People would, they take the advertisement pamphlets and all that, but getting people to actually do the work was more difficult. And so, yeah, I think it's definitely doable. It requires a really serious PR outreach. Maybe some of the other panelists know more about iNaturalist.
Yeah, I would add that I think this is really important because it helps us get to better understand distributions, both of native and invasive species. And, I don't know personally how to do this, but I know that there's ways to do like web scraping where you just kind of go to these sources like iNaturalist and EdMaps, and you can get a bunch of data in a really short period of time. And so, there are people that have, really good computer science and data science skills that are really valuable because they're learning how to integrate all these different platforms together to, enhance our understanding of species biology and distribution.
Thanks very much, Angela. Go ahead, John.
Yeah, I would have to agree with Angela. I scrape the heck out of these things because the old distribution information, especially on plants, was very inaccurate. It might have been accurate for identification. They nailed that, but distributions, if you're looking at data from the 70s and 80s, they'd say it was in this county, or township range, nothing very good. This is a wonderful way, when they're linking their photo to their latitude, longitude, putting it up there, asking for corroboration on their ID, to get so much data that is pretty darn accurate. I use it all the time for going out to find native and invasive samples. in a way that, I can't afford to, we can't afford as an agency to go mine that, get that data, but we can sure mine it pretty easily. And I think there's such a great advantage that the people get involved and start learning more because they have their interest, and they get to interact with people who are experts on these platforms. And that really brings up the citizens' level of science and love for science. So it all spirals up.
Yeah, there's actually been a lot of engagements in terms of citizen science on the environmental DNA front, because it's as a technology, it's kind of built for that sort of thing. There are a lot of little projects going on here and there, but some countries are taking it pretty seriously. I know New Zealand has a nationwide program to engage particularly Maori communities in using eDNA to start better understanding what's in their aquatic resources, the biota that's there. And it's been pretty successful, I think, at engaging those people and getting them more interested in the science and actually starting to drive the science more into the mainstream, which is only going to make it better, I think.
Excellent. Any further comments on that question? Thanks so much, everybody. We do have a question from Donna from the floor. And so she's asking is the adaptation of plants toward where water is available. What can you see happening as to their relocation to higher altitudes or latitudes? How long do we have to see the change of ideal habitat zones for native species so they can ward off either invasives or pathogens which will impact their ability to reproduce and fight? Oh, sorry, that just shifted down for their habitats. So I think the question in there is, what can we see happening as to the relocation of invasives towards higher altitudes? And is there a lag time between native species being able to compete with that?
I mean, it depends is the unsatisfying sort of ecology answer to that question. There's definitely work being done on this. I've seen some stuff out of Switzerland and stuff where they're very interested in the same kind of thing, like colonization of widespread North American invasives and colonizing there. And it just really depends on the species. What's its method of propagule dispersal? Like, how do the seeds move? The recipient communities, how disturbed are they? In the case of there, you got a lot of grazing going on, or a lot of roads, skiing access, that kind of thing. If it's heavily disturbed, as we know from our experiences over here, disturbance is ideal for many of these invasive plant species. And I will just acknowledge her point in there about the moisture availability changing independently of temperature as you're going up, especially in elevation, but also latitude. So it's definitely something to be aware of.
Yeah, I think in Alberta and BC and other parts, we'll see that northern shift in communities. So grasslands in Alberta will start encroaching on the forest, and we're already seeing some signs of that at the boundaries, and we'll see even more signs of that happening in the 50-year sort of time frame. So I think what's really important is that we provide corridors for native species to migrate. So things that aren't fragmented and cut up by roads and trails and all of our activities. So corridors and refugia will be really important in aiding species migrating to their new climate niche.
Very good point. Thanks so much, Shauna-Lee. Thanks, everyone. I think I want to go to the next question, which is just, in addition to sessions like we're having today and the research forums that we've hosted in past and things like the research hub that we demoed earlier that allows some interconnectivity between practitioners and researchers, are there more things that could be done to open that dialogue up in a better way, or do you have thoughts on things that we could do to better improve that dialogue?
It's something I've thought a lot about, and people have written loads about it. They sort of call it the knowing doing gap, and some folks say there's an absence, or science could be more involved in governance issues, and it's really an interesting subject, and I don't have a great answer, but I think forming relationships through forums such as this one is really important. if I'm a practitioner and I have a question, if I have a go-to science guy, I should feel comfortable to contact them. And that's just how I sort of see it playing out through the formation of relationships and ties, which can be hard sometimes because scientists are introverts, but I don't know.
Yeah, that's a very good point. And certainly that was the driver behind the creation of the Research Hub, is trying to provide a bit of an interface for that discussion and again, events like today. So, but it's an excellent question, and I think something that we all can work more on. Was there anybody else from the panel that wanted to offer any comments on that question? Go ahead, John.
Yeah, I think the Research Hub looked a really great idea, you know, and we have a few things, like we have like quarterly calls on biocontrol of weeds in the West. You know, Carol Randall puts that on from the Forest Service. Those are really, really good opportunities where scientists are listening in, land managers are talking about their problems and their issues. And it's great for getting that information flowing back to the researchers. You know, you have enough of us putting our papers out or giving a talk, but I find that these kind of coffee hours where we can hang out are really, really bringing in a lot of engagement and better questions. And so are things like iNaturalist and EdMaps, where you have an interaction with people, with experts on whatever they're talking about right there. So it's getting better. I can't think of any novel ways beyond that would make it super fantastic.
Excellent. Thanks, John.
Yeah, I'd like to second what Shaunalee was saying about relationships. I think that's really important. I mean, as scientists, I think we like to think that the science will kind of carry the day, that if you provide good information to people, that will be enough. But I think in reality, it's the relationships that make a difference. It's that trust that is built between individuals that's maybe even more powerful than good information. And the other thing that strikes me sometimes is that I think the reward systems are different in the two worlds. And sometimes there's a mismatch there that's unfortunate. I mean, scientists, sometimes we're working on, you know, cycles with the time frame of a grant proposal or something like that. And, you know, our rewards are tied to publications and maybe not to the same sorts of things that managers really care about. I think this is changing for the better. And I think organizations are recognizing that in order for scientists to get engaged in the ways that make a real difference to managers, they need different types of rewards in their careers. And hopefully that continues to improve.
And I'll add one more: publish in open access journals.
Yeah, very, very good point. Thank you so much. We have another question from David, just for panelists that have a background or specialty in terrestrial species. There's a question about what about increased fire frequency and intensity, and how does that promote invasion of species?
I'll take a first stab on that one, I guess. Yeah, it's a huge issue. So if fire frequency is increasing, and certainly there's a big one that just took off last night just outside of Penticton. Yeah, a lot of it, that's a disturbance, right? And so you get if the intensity is high enough to, you know, damage those locally adapted, native fire adapted species to the point that it becomes an actual disturbance. You just have open soil. You're getting colonization of a lot of invasive species. I know it was a real concern near Cache Creek, I believe, when there were some fires there in the past few years with things like spotted knapweed colonizing heavily afterwards, and ranchers and rangeland tenure holders and people being very, very concerned about these rebounds and the fire kind of being a core mechanism of that for sure. Yeah.
Thanks very much, Dave. Thoughts? Go ahead, John.
Yeah, along with fire control being a huge transport issue that we see. I mean, we all see a lot of line area where the fire truck was parked, unfortunately, through no fault of their, you know, it's a difficult situation. It's a big spread over long distances, which are the critical movements.
I would quickly add that actually that has impacts for freshwater systems as well. Potentially, when they're picking up water, they could be picking up water from an invaded system and moving it to around to a different system and picking up organisms and moving them. So yeah, it kind of crosses, it's an interesting problem that crosses boundaries.
Very interesting, very good point. Thank you, Angela. I think we'll take one more question from the group. So we have a question from Shauna to the panel, just going back to John Darling's presentation and the slide depicting the generalized invasion curve. And the question is really, where do we focus our efforts as land managers when we already have species, including hawkweeds, cinquefoils, St. John's wort, that are significantly altering high-valued ecosystems? Where's the balance in the use of our limited resources between early stage eradication and protecting more high value resources? So just a question on sort of direction to managers on prioritizing.
I'm not sure how to answer that. I feel a little hesitant to tell a manager what to do. But what I know is that all of the research that I have seen indicates that prevention is by far the best bang for your buck. And there's some additional research that indicates that it recognizes the difficulty of selling prevention as a method. You know, when you're doing eradication or control, it's easy to see the gains that you get for every dollar spent. When you're doing good prevention, the gain is that nothing changes. And so it's difficult to see that, but every modeling exercise, every bioeconomic model that I've ever seen shows that prevention's where you want to be spending most of your money. I mean, obviously your portfolio has to be diverse, and there are things that you're going to have to control, but in terms of investments, prevention's the way to go.
Thank you very much, John. Go ahead, Shaunalee.
Particularly with respect to climate change, I think targeting those nascent invasions, those early stage invasions after those extreme events. So if a fire goes through, if a flood goes through, get those high impact species that are just starting to invade where they stand to threaten biodiversity or agriculture. That's my sort of big, fluffy, cloudy advice.
Yeah, that's an interesting point. So I recently saw demonstrated a tool developed by our U.S. Geological Survey, where they're actually trying to forecast invasions associated with things like flooding events. So they basically look at how those flooding events might cause interconnectedness between watersheds that previously were not connected. And they have databases where they know which invasives or known invasives are present in various watersheds. So they could, you know, presumably generate early warnings around these extreme events to indicate where you might be worried that invasive species X has spread beyond where it previously was. So those kinds of tools might be helpful for those sorts of events.
I sort of view it as a double whammy effect. You have this disaster and then you have this species that comes in.
Absolutely. Thanks very much. We are sort of at the close of our time for the panel now, so I just want to take an opportunity to thank each of our speakers very much for spending time with us today and sharing your wisdom and engaging with the group through their questions. I've learned a lot. I hope that the rest of the group has also learned a few things that they'll take back into their work and potentially their research, and hopefully made some connections as well. I'll invite our speakers to turn your cameras off. You can get out of the hot seat now if you like. We do have one last interactive activity that I'd like for us to just do together before we wrap up today. So we will have a link that'll be shared on your screen in just a second, and we'll also share it in the chat box. But we'll invite you to go back to your browser, whether you did the previous one on your phone or on your computer or tablet, and just enter in one word about how you're feeling after having attending the Invasive Species Research Mini-Symposium today. So you'll see a couple of people have started dropping words in there. We're just going to create a fun little word cloud just to get a sense of where everybody's at after doing the event for today. So we'll leave that open and you'll see that change in real time in front of you. So take a minute and do that. It does ask for your name. You can skip past that if you prefer not to. The information isn't shared, it remains confidential. So it's popping up as we speak. So we'll let that populate and I'll just share a couple of thoughts with you. As it came up during our panel, just a reminder to anybody that's not already a part of the Research Hub, we invite you to become a member and to join and to connect with other researchers and practitioners across the Pacific Northwest. That's the purpose of the hub, and we hope that it provides value to you. So we ask that you can join that. The link was provided in the chat. You can also join that directly by going to the BC and Invasive Species Council of BC website, and you'll find the link there. We also very much would like your input on the event today to help us improve our virtual events and to see how everybody felt about the content that we shared today and how you enjoyed the session. So once we finish the word cloud, we also will be posting just a quick link for you to do a survey. If it's possible for you to do that before you leave today, it would be great, just so it's fresh, top of mind. If not, we'll be sending the link out afterwards. And the last little reminder is that to support the work of the Invasive Species Council of BC, if you're not already, we invite you to become a member. It's easy to do. It provides you with some neat perks and resources and lots of great information that gets shared out through our network. I think having said that, I just want to take a minute and have a look at the word cloud that's popping up. This is great. So some of the big, so when a word is shown larger in the word cloud, it means it was entered by more people proportionately. So it's displayed as larger on the screen. So some great action words. So inspired, informed, curious, motivated, challenged, enlightened, productive, but also a little bit apprehensive. You know, certainly there's some, you know, a little bit overwhelmed. So I think there's probably a balance between all of those pieces. I do just want to thank all of you for joining us and for engaging with us today. And I'm going to ask Crystal Chadburn to do some final concluding comments before we wrap up. So Crystal, over to you.
Great. Thank you so much, Jody. So it's been a real pleasure to be part of this morning's Invasive Species Research Mini-Symposium. And I'd like to start by offering a special thanks to John Darling and John Gaskin for their, doctors, for their outstanding keynote presentations. Thanks also to our Pacific Northwest experts, Shauna Lee Chai, Angela Strecker, and Dave Ensing for bringing their climate-related research projects to us. And finally, again, to all 5 of you for contributing to the engaging panel discussions. That was really informative. Thanks again to the province of BC for their financial support of this event, and a huge thank you to the members of the Research Conference Advisory Team, who I know firsthand have contributed lots of time and energy to the planning of the Research Conference and this mini-symposium, and of course also to the Research Hub and the Research Roadmap. So we would like to also acknowledge the ISCBC staff who have been busy behind the scenes and have helped move this mini-symposium through very, very smoothly today. Lastly, on behalf of the Invasive Species Council of BC and its Board of Directors, we once again thank all of you for joining us and helping make this event a success. We hope you have learned something new or found a potential new professional connection that you can follow up with. We look forward to reconnecting in the future, hopefully in person at the maybe 2021 Research Conference. If we can host it, we will be doing that in person. And of course, in the meantime, we can all connect together on the Research Hub. So have a great rest of your day, and thanks again. Bye-bye.